let me really start off by acknowledging the traditional custodians of the land, the Gadigal people of the Eora Nation, and I really want to pay my respects to their elders, past, present and emerging, and extend that welcome to any Aboriginal or Torres Strait Islanders who are here today. So SPHERE, the Sydney Partnership for Health, Education, Research and Enterprise, is what we're all a part of here today. But what you may not know about SPHERE is it has this name given to us by the Darug people, Maridulu Budyari Gumal. And this really embodies why we're here today. Working together for good health and well-being is what it means. And so I really want us to really capture that as part of what today is. It's a workshop. The idea is not for those who are speaking to tell you how good they are and how amazing their research is, but for us to get a sense of all of the different aspects of the therapeutic sort of translation pipeline, and for us to be able to work together to work out better ways to do that within SPHERE and utilising the resources of SPHERE. Now, what are the resources of SPHERE? Well, here are a whole lot of the institutes that are part of SPHERE. So you can see here throughout the day we'll have speakers, we'll have panel members. We've got Mary Bebawi as the facilitator for the day, and thank you very much for taking on that role. And really we are not about one of these partners, we're about all of these partners and trying to utilise the full spectrum of SPHERE to be able to drive therapeutic development within the Sydney area. I want to particularly thank the Cancer Clinical Academic Group, the Faculty of Medicine at UNSW and the Translational Cancer Research Network for their support of this event. This is a free event. Because of that, they're providing all the resources here to film it and make it available not just to the 90 of us within the room here, but also across the whole SPHERE network for the future. And today we are particularly thankful also to the other conveners. Phoebe has really done an amazing job. Greg and Steve Palmer, really we couldn't have got all of the speakers and the panel members and everything together. But almost most of all, Tanya Ward, what an amazing job you've done putting all of this together. She's been the one who has set up this beautiful room and really everything today. So thanks to Tanya for that. So I've been given the task of welcoming everyone, which I've now done, and I'm going to give a bit of an introduction and overview based on some of my research over the last sort of decade. And really, in terms of introducing, what we're doing here today, I thought about trying to get one of these convoluted diagrams here where, we talk about what the targets are and how we go about making a drug. But, these are really some of the steps that, I wanted us to highlight today. We've got talks about identifying and validating targets, whether that's a target protein or a cell function. And so we're starting off with my talk and Francis, talking about some of that. We've got the whole middle section on high throughput assays, medicinal chemistry. And whilst today the focus is really on cancer therapeutics and medicinal chemistry, small molecule inhibitors, a lot of what we're going to talk about today will apply across other areas, whether that's biologicals or nanomedicines, etc. And hopefully in the future we'll have some more events that cover off some of those areas and go beyond cancer. And that's certainly something that we're interested in. We're going to talk a bit about preclinical models, and we've got Lisa Horvath talking about that also in the first session. And then in the final session, and I really hope everyone sticks around, because you'll see throughout the talks, through my talk and numerous other talks, how much we need to be thinking about everything that's in this third session right up front. If we're not thinking about what a target market is, what are the regulatory processes we need to cross through, how to get things into clinical trials, we're not going to succeed, we're going to fail early. And so, if we can go through preclinical models, clinical models, the ultimate thing is to cure the disease. That's what we're all here for. And hopefully today we can talk about ways to better do that across the SPHERE partnerships. We're not specifically going to talk about funding today, but ultimately the goal of something like this is to generate new collaborations that will be able to be successful in things like MRFF funding and some of these big funding opportunities that come up. So, we're hoping to be able to try and get some smaller grants, maybe through CAG or TCRN or Concert or Kids Cancer. So through some of these agencies to be able to get some pilot studies underway, and that's the sort of thing that we could get access to then ultimately leverage bigger funding opportunities in the future. So what have I done? First of all, I do have a disclosure, so I'm founder, CSO, shareholder, and have had research funding from the Medical Research Commercialisation Fund, UniSeed, and we partnered with CSIRO, and so that's what I'll be talking about today, is how we founded Metablock Pharmaceuticals. So the idea that we came up with, so my research over the last sort of 12 years has focused on cancer metabolism, trying to understand how cancer cells take up and use nutrients. And so one of the transporters that we came across during that time was ASCT2, which is a transporter that brings in glutamine among some of these other amino acids. Now this is upregulated in many cancers, and you can see that it affects a lot of different pathways. We've got purine and pyrimidine biosynthesis, so DNA and RNA synthesis, TCA cycle, protein synthesis. Some of these other amino acids are involved in one-carbon metabolism and antioxidant redox. So the idea we had was, well, this is upregulated in multiple cancers. If we can block it, we can pretty much cut off cell growth and survival at multiple different pathways. So what sort of evidence did we need to be able to think about this? What we did was, first of all, to look in prostate cancer. That was the disease we were working on at the time, and we were able to show that very nicely we had expression of ASCT2 at all different Gleason grades. We showed that it was regulated by the androgen receptor, so it actually decreased after neoadjuvant hormone therapy, but came back in castration-resistant prostate cancer. So we've got a protein that's highly expressed in prostate cancer, both in the early stage, but also in late stage disease. So that was a big tick to start. So we can see here in a preclinical model, this is an LN-cap xenograft model, we have high expression. It's androgen regulated, so it decreases after castration of the mice, but comes back in castration-resistant prostate cancer. So it's expressed in the models we're interested in as well, and you can see here that when we knock it down with an shRNA, we very nicely reduce tumour growth. So that was the idea. Okay, great, we've got a target, it's highly expressed in prostate cancer. If we knock it down, it decreases growth. This is exactly what we want to do, but there were no drugs to be able to target ASCT2. So the way we approach this, and you'll hear throughout the day how different people have approached their drug screening. We did ours using in silico models. So basically we made a structure of ASCT2 based on a bacterial transporter and then ran a drug library from specs. So this company has hundreds of thousands of compounds that can be ordered for $20 for 10 mg, and so you can just run your library through this model, come up with a handful of compounds and then just order them. So we ordered 47 different molecules and very nicely got one hit, which we named very innovatively Compound 19, because it was the 19th compound that we looked at. And at this point we thought, well, this is something now we might be able to try and get some funding to really take forward as a therapy. On the side, we had also been working in other cancers, and one of those was triple-negative breast cancer, and we're just waiting again for the computer. So we showed that ASCT2 was highly expressed in triple-negative breast cancer patients. Here is the data, so you can see really nice expression in the majority. Only one triple-negative breast cancer patient didn't have expression of ASCT2 protein, and then we also showed that ASCT2 knockdown worked similarly. But nicely, we had a doxycycline inducible version, so we could allow tumours to form and mimic actual inhibition as you would with a therapy, and we showed really nice survival benefit. And in fact, a couple of mice we culled that didn't seem to have tumours anymore. So when we went and decided to try and get funding through the Medical Research Commercialisation Fund, we were really talking about prostate cancer. All of the work we'd done with Compound 19 was prostate cancer, and we went to them and we said, this is what we've got. We've got a prostate cancer therapy that might also be applicable for triple negative and melanoma. And so we put in our pre-application. So I'm going to just talk in the next few minutes about some of the things that we learnt through that process. So one thing they really wanted from us was very clear biology, where you have a niche expertise. Now, fortunately, not many people care about amino acid transporters. So we were one of the world experts in that area, and so that sort of fit the bill. But they also like to have that supported by other people's papers, because if it's just your lab that's shown it, you know, maybe you've got some unique models that don't apply everywhere. So fortunately for us, our oncogene paper was actually scooped by a cancer cell paper. Not often you say that, but where they were able to show that SLC1A5, which is ASCT2, is highly expressed in triple negative breast cancer, and if you have high ASCT2 and triple negative, you do much worse. So this was really perfect for us to be able to say, well, you know, this is actually a good target, and someone else showed it, but you know, we've got the expertise in this area, and so obviously you need some kind of unique test, and or a lead molecule or a hit molecule that needs to be further developed, and you know, which has not been disclosed, which we had. We'd never talked about compound 19, never shown its structure or anything, so that was good. And we had a tritiated glutamine uptake assay, which was a 15-minute assay done in a 96-well plate, where we could, you know, really test these therapies in a semi-high throughput fashion. So one thing that really sort of surprised us is they were really keen to know all the weaknesses. Now, when you put in an NH and MRC grant application, you tell your story, and it's a beautiful story, and you don't really focus on the weaknesses, but they want to know, tell us, what are the resistance mechanisms? How is this going to fail so we can work out how to make it work? And I really love the honesty of this approach, where you just go in and you say, I'm a basic biologist and I know nothing about anything else. I don't know about medicinal chemistry. I don't know about clinical translation. I don't know about preclinical models. And so they said, well, here, get in touch with these people. They'll help you with this. And they got us in touch with all of the people that were necessary for us to know how to take this forward. And that's where Sphere, I think, is really going to be important for us as we're thinking about how we can take molecules forward. We've got everyone we need within this room, within our various partners, to be able to have the expertise across all of these different areas. So Elgene and Alex had these triple negative breast cancer patient-derived xenografts, and we were able to show that 5 of 9 had screamingly high expression of our target, and that gave us some preclinical models to be able to test in. The potential market was a really interesting one, because we went in as prostate cancer and they said, well, you know, Lisa, I know you say it's fantastic, but they said triple negative breast cancer, that's a much better market. You know, short 5-year survival, you know, poor 5-year survival rate, that's going to be our target market. Forget prostate cancer, we can come into that later, but that was, everything was taken forward in triple negative breast cancer because, you know, that's where the market that they saw was. And one of the big things was they wanted someone, they talked about this two-head test, you know, can we work with you? Are you some kind of two-headed monster where you're just, you know, going to want to control everything? They want to be able to work with you, that you would take advice and be able to move these things forward. So we managed to get through all of the various different pitches and got our first tranche of funding, which was really partly because I think being a very early hit, CSIRO came on board for all the medicinal chemistry and gave in-kind funding for that. So we started Metablock Pharmaceuticals. Uniseed then came on board with some funding as well. So we had our first tranche of funding, had some milestones, hit those milestones. So this started back in 2016. We had our tranche 2, and unfortunately earlier this year we didn't hit our milestones, so we're kind of in limbo on that at the moment. But we can discuss more about that in the panel if people are interested. And just one final point, I think, that we're not I think Mark Cowley will talk a little bit later about some biomarker, genomic biomarkers. But I think we need to be trying to think about biomarkers a bit more in this work as well. So we showed that ASCT2 is actually highly expressed in luminal A breast cancer and triple negative breast cancer. But if you inhibit it in luminal A breast cancer, it does nothing. So what we've come up with now is some metabolic biomarkers that really distinguish triple negative from luminal A, which are really these various different pathways which are not being highlighted right now. And what we've been able to show also then, if you look at those biomarkers in prostate cancer, you can show that there's a certain proportion of prostate cancer patients that also have those. And so these could be a good biomarker for us to know which patients to treat with ASCT2 inhibitors in the future. So I'm going to finish there while it's slowly catching up to itself, and we will move on to the next talk.
So thank you to the organising committee for giving me the opportunity to facilitate today. And this is a very exciting field. It's an exciting area for me. It's highly relevant. And today we're really being taken on a drug discovery journey. And we're starting right at the beginning of that journey with target identification and validation. So I'd like to introduce today Dr. Frances Byrne, who will be taking us through her research, but more importantly, really what she's learnt and what advice she can give us. So thank you.
I'll talk about a project that really kind of started when I was doing my first postdoc at the University of Virginia in the US. And contrary to what the program says, it's not too much about target identifications, but it's more to do with actually a phenotypic screen that we started. So typically we know that there's a lot of target-based drug discovery approaches which have led to fantastic successes in terms of cancer treatment, in particular Gleevec for chronic myeloid leukemia. But we also know that there's some really mixed results with this kind of approach, in particular the treatment of melanoma with this drug, verafenib. We know that patients can respond really well initially, but then they become resistant quite quickly, and so this is a huge problem in cancer treatment. So with that in mind, and with kind of information based on previous literature, we know that phenotypic assays instead identify drug leads and clinical candidates that are more likely to possess therapeutically relevant molecular mechanisms of action. And so we really wanted to kind of look at this approach instead of a target-based approach for identifying novel anti-cancer agents. So the rationale for the way that we wanted to do this drug screen is that I have done a lot of metabolic and metabolism-based experiments in a number of different types of cancer. And so this is just showing the results from one of the cancer types that we work on, which is endometrial cancer. So I have a panel of endometrial cancer cell lines shown under the red line, and then 2 non-cancerous cells. And we did some radiolabeled glucose tracer experiments where we can measure glycolysis. And so this is known as the Warburg effect in cancer, where they metabolize a lot of glucose. And we can see that the majority of the cancer cells have much higher rates of glycolysis than on the non-cancer cells. But what was even more striking was that when we measure glucose oxidation, so that's the metabolism of glucose to carbon dioxide, we actually see all of the cancer cells have lower levels of glucose oxidation compared to the normal or non-cancer cells. So from this phenotype, we wanted to kind of exploit this, and to do this, we wanted to exploit this phenotype for a drug screen, really. And so we know that most cancer cells exhibit lower glucose oxidation than cancer cells, but also a lot of cancer cells are more susceptible to destabilization of redox balance, and that's generally because they have higher levels of basal ROS than normal cells. And so one way that we can exploit this is potentially through increasing oxygen consumption rate, which is an indicator of a change in cell metabolism. So these are just some details about the drug screen that we did. Obviously, we're not talking about hundreds of thousands of molecules, which are, can be done with through different means. Through Greg Arndt would know very much about this, but we have a much smaller library. So this was done by hand by a student in Kyle's lab. So we had more than 5,000 compounds that was a maximised chemical diversity library. that we screened in biosensor plates, and basically an increase in fluorescence was an increase in oxygen consumption. We had a positive control, and then we had a kinetic reading. And so basically from this screen, we had 25 hits that increased oxygen consumption rate. And then I took these 25 hits and screened them against a panel of a number of different types of cancer cells, as well as normal or non-cancerous cells. And so from this screen, our best molecule was the molecule that we've named BH10. So I'll just give you a little bit of an overview about what BH10 does and how it works. So this is just a snapshot, just looking at one single dose of BH10 in all these different types of cancer cell lines, looking at cytotoxicity. We have also a number of normal or non-cancerous cells as well, mainly shown in blue. And what we can see is we do get various levels of toxicity. Not all cancer cells are sensitive to this agent, but we do see that the majority, compared to the normal cells, are sensitive. So then I'll kind of take a step back and talk to you a little bit more about how we went about developing this molecule and potential novel molecules from this screen. And so this phenotypic screen and the identification of the hit molecule really was quite quick. But what's been going on since about 2013 has been a number of different types of experiments to kind of really move this project forward. So I did a lot of benchmarking. I've compared it to more than 20 different types of drugs. What I'll talk to you a little bit about today is some of the mechanistic studies or the mechanism of action experiments that we've done to try and identify a drug target. We did drug formulation, just trying to get it into a solution that we can give it to mice with. We've done pharmacokinetics experiments, in vivo testing in xenografts. We've also been working closely with Dr. Naresh Kumar to identify or create novel molecules and doing a lot of structure-activity relationship experiments. His students have been really fantastic, and they've developed more than 30 molecules that we've been screening in vitro and also doing pharmacokinetics on. So talking about the really easy task of trying to identify a drug target if you're doing a phenotypic screen. So this is really probably the major challenge that you'll have if you do a phenotypic screen. So these are some of the approaches that we took to try and find the target. We did a number of bioinformatics approaches using data that has already been published related to mutation status and gene expression using the Cancer Cell Line Encyclopedia from the Broad Institute. We did drug-target interactions where we biotinylated the molecule, tried to look for interacting proteins. We've done knockout of potential targets using CRISPR to look at drug sensitivity. We did a kinome array. We've done a reverse phase protein array looking at phosphorylated proteins. I've done a number of different types of in vitro tests looking at ways to potentially rescue the cell death phenotype caused by BH10. But basically this led to a lot of frustration and not an answer. And I guess looking back, it is important to do these experiments, but at the end of the day, the phenotype really is what you'd be wanting to look at. And it's known that a lot of drugs out there, their targets haven't even really been validated yet. So it's not the be all and end all. So this is just a quick snapshot of how we think this molecule is working. I won't go into the details of it, but basically the methods that we did use that kind of were helpful were metabolomics, using radiolabeled glucose and other nutrient oxidation assays, stable isotope tracing, enzyme overexpression assays, and oxidative stress assays. And we really kind of did all these mechanistic studies and found out that it's probably working via inducing mitochondrial oxidative stress. So this is kind of talking about some of the methods and a lot of the things that we do in our lab on a routine basis. And so if anyone's interested in doing metabolism work, you can also have a chat to us later. So this is really the way that we kind of found out a bit more about how this drug was working. So I guess the other challenge that we've really encountered, which has been probably the major hurdle of the project, is trying to test this molecule in vivo. And so these are some pharmacokinetics results that we have, where we injected the mouse with 10 mg per kg of our drug in this captazole solution. But the problem with this is that basically with our molecule, we have a very short half-life in plasma. We're getting very low concentrations in circulation, and potentially it's not orally bioavailable. So this has really limited how we can test this molecule in vivo. So what we've been doing with Naresh has been developing new and improved BH10-like molecules, and the way we do this is using very talented honours students to create the molecules, and they've also been doing some of the screening for us in vitro. So we screen them for sensitivity in cancer and non-cancer cells. If they lose that selectivity, it's canned, and if it's retained, then we keep going with that molecule. We then do pharmacokinetics in mice. If it has a short half-life, or is toxic, or is not orally bioavailable, we can it, and then the ones that have those desirable features we'll keep pursuing. And then we hope to use these and test these in preclinical cancer models, and then hopefully if they do have good activity, we can kind of look forward into further drug development. And really this is an iterative process, and so at all stages we're getting more and more information based on the different structures. So just quickly to show you one of the new molecules that one of Naresh's students developed. So the cool thing about this molecule is it's not in the public domain, so it's potentially patentable. We see that it has a longer half-life. We can also detect it in plasma if we give it to the mouse by gavage, which means that it could be potentially orally bioavailable. So this molecule has more desirable features, at least in vivo, compared to our original molecule. So what are the lessons learned? So importantly, that initial screen was great. It identified a good lead molecule, but the poor pharmacokinetics in mice has really limited its further testing in preclinical cancer models. Future work, which is the big lesson that I learned from this project, will actually prioritise pharmacokinetics pretty highly, because at the end of the day, if you can't test it in a mouse, you're not going anywhere. So for us, that's probably the number one thing that I've learned, is to be looking, if you're doing these drug screens, to make sure that you can get it into a drug formulation that it has good properties in mice before you really kind of look, maybe then go and do mechanistic studies a bit later. And then also I'm hoping to, in the future, if we do find a good candidate, maybe employ a different method. And there's been a good paper out recently, or maybe in the last year or so, where they use CRISPR-Cas9 mutagenesis experiments to identify a drug target. And I think this has also identified a lot of new targets for current drugs, and so this could be a potential way that we go about that. So I'd just like to thank all our collaborators with this, particularly Dr. Naresh Kumar, for really kind of helping us kick off this project, and Beverly, who did that initial screen, and and obviously the funding from the Cancer Institute New South Wales and the Hope Funds when I was in the US. So thank you.
So our next speaker is Professor Lisa Horvath, and she'll be speaking to us about laboratory and preclinical models. So welcome, Lisa.
So I'm going to take you through 3 stories, and I'll start off by explaining that I'm a weird hybrid. Not only do I have enough, that's only the bits of my affiliations. I've spent 20 years sitting between University of New South Wales and Garvan and Sydney University and RPA Chris O'Brien Lifehouse. So I'm a hybrid from a university, and I'm a clinician scientist. So I run a wet lab, but I also run a clinical trials unit and a clinical practice. So I'm going to tell you 3 stories of how our group has tried to get from target identification actually biologically into the clinic, and I should have called this 2 steps forward, 1 step back. I've learnt a lot in the last 5 years. So these are my disclosures. And I was recently, actually only at the end of last week, in a review that talked about this translational paradigm and wants to rename it from bench to bedside, bench to patient. And I rather like that because bedside sounds a bit too impersonal, and really what we're wanting to do is to get this into patients. So these are going to be little vignettes. I'm going to start off by talking about localised prostate cancer and then move on to metastatic disease and how we've tried to address the issue of new targets across this paradigm. Prostate cancer has a very long natural history, and proof of concept studies for your target are incredibly important. So story number 1. Story number 1 involves Lisa Butler at the SAMRI over in Adelaide, who has been doing some beautiful work for a long time using cancer explants, where they take frozen tissue out of a prostate at radical prostatectomy for localised disease. They put it on a sponge, a gelatin sponge in culture media, and it allows them to do up to 8 parallel experiments on the same piece of tissue, looking at different looking at different targeted agents. And this is really the work of Lisa Butler and Maggie Centenera. So a few years ago, Lisa and I were chatting in Adelaide, and she was showing me this really beautiful work where she was looking at a variety of HSP90 inhibitors because they're chaperone proteins and could be used to get round drugs like enzalutamide and abiraterone in prostate cancer. And you can see from the top graph a beautiful one showing that Ki67 goes down very nicely with AUI922 and another HSP90 inhibitor, and the bottom one is cleave caspase 2 looking at apoptosis. So this looked very promising. However, and there's been mouse model works and things like that, so on the back of a paper napkin we wrote a proof of concept study that amazingly got funded. The idea that we would, I won't put it yet, the idea that we would treat men for a month before radical prostatectomy, look at their prostates, have the biopsies from beforehand, have the tissue from after, and be able to look at biologically whether we were hitting the target, causing apoptosis and decreasing proliferation. And this all seemed very nice, and we got the grant, and we wrote the protocol, and then we hit the complication. The complication being that 3 weeks before we were due to open the study, all authorised, all ready, all sites in place, Novartis, who was the company who owned AUI922, said, "Oh, we've decided to stop development. We've sold the compound." I'm sorry. What do you mean you've sold the compound? Oh, actually it wasn't ours. We were licensed it out of this little company that had never made human-grade drug, and we folded the whole program and we're not interested anymore, and we don't have enough drug in the whole of the world to do your study. You can see my frozen expression, still thinking about it. And I'm like, right, I have $600,000 to do this study. I have funders, and by the way, some of them are the PCFA, and I'm prepared to tell them you guys aren't very nice. What do we do now? So we went back and we looked through other explant data, and we identified that actually Lisa and Maggie had done a study with Karen Knudson's group looking at palbociclib, a CDK4, CDK6 inhibitor, and had shown good inhibition in the explant models. So this is how we then went back and go, well, this drug isn't from Novartis and they're currently feeling really, really guilty. So we went to their back catalogue and realised they had ribociclib, which is also a CDK4, CDK6 inhibitor. I can't put it up here because it's unpublished, and if I want the slides to be allowed out, I can't put it here, but we found exactly the same data with ribociclib, and we did that really fast and said to Novartis, okay, you've put us in this spot, now give us the drug. And they did. So at this point where we were up to several extensions on our grant, But what we now have open is a phase 2 trial looking at the pharmacodynamic effects of ribociclib. It's in high-risk localised prostate cancers, and we treat men for 3 weeks prior to radical prostatectomy, and we have 6 patients already recruited onto the study, and we're opening across 6 sites around Australia. We've got 2 open at the moment. And what will this yield us? The primary objective is to look at the rate of Ki67 proliferation decrease. Because they're high-risk prostate cancer men, all of them have Ki67. We can find Ki67 index in them. But we're also going to look at how are we affecting CDK4/6 inhibition, apoptotic cell death, and a variety of other markers. So what will this do for us in the end? Basically, what it's going to let us know is, are we actually getting biological activity? As you saw from the natural history of prostate cancer is many, many years. It's great. I have a long relationship with my patients. But it's horrible if you're a researcher, because it means if you want to look at biochemical relapse and see if your drug affects it, it's going to take you 10 years to know whether it's effective. So the best thing you can do is screen for biological activity before you put it into the patient for that. What's going to be really interesting is, does this validate the explant model? Because in prostate cancer in particular, the mouse models, I apologise to anybody who uses them, are dreadful, and they do not represent the patients. And so we really do need good models that can predict for us what's going to work in prostate cancer. So if this validates the explant model, that really says the explant model might be a very good preclinical model to use. So that's the positive. I'm putting the negative one in the middle. Then we look at metastatic prostate cancer. So this is going to be very short because it was many, many years to not get a long way. For many years, Roger Daly and I have been looking at FAK inhibition and docetaxel resistance, docetaxel being the major drug in metastatic castration-resistant prostate cancer. And this is some work done by Brian Lee, who was a joint PhD student, showing that a drug called defactinib, which was a drug owned by Pfizer, a FAK inhibitor, does a very nice job in resensitising PC3 and DU145 prostate cancer cells, androgen-independent cell lines, to docetaxel. We also showed it in xenografts, that this drug worked very nicely. And again, we used the explant model that we've now set up in our own lab. VS6, by the way, is just one of the names for defactinib, the FAK inhibitor, and showed that we got a significant increase in cleaved caspase 3 in the prostate cancer explants. So here we go. We've got beautiful data. We've got cell line data. I won't show you. We've got 101 bits of functional data showing that we're inhibiting FAK, we're killing cells. It's all beautiful. We've got xenografts, we've got explants. You would have thought this was a slam dunk. Except the drug was initially developed by Pfizer. They gave us the drug on an MTA. By the time we'd finished the experiments, they'd licensed the drug to a place called Veristem. Veristem then used it to run a couple of really big clinical trials, one of which was in ovarian cancer, looking at taxanes and looking how it was synergistic with taxanes. But one of them, which was their big flagship, was on mesothelioma that went down in flames and bankrupted the company. It's currently just out of receivership, but they're doing really small clinical trials, and I can't get the drug for love or money. So, despite having every model I could think of, there is no way this is going anywhere at the moment, but I'm leaving it in my back pocket, and I live in hope. Finally, I want to take you on my last story, which is less about the target and more about how you target the biomarker. So we've, in the last 5 years, stumbled into looking at lipids in the circulation, which, as many of us know, many of the things we end up in are pure serendipity. This was my postdoc who'd done her PhD in metabolomics and said, I went to this interesting talk by a guy at the Baker called Peter Michael. Have we thought of doing lipidomics in prostate cancer? And I went, oh, well, let's give it a try. The first thing she pointed out to me, much to the horror of several of my colleagues, is that there's much more lipid than DNA in the circulation. And I work a lot with people looking at circulating tumour DNA. So what we did is we took a cohort of patients, 96 patients with castrate-resistant prostate cancer, and we did a lipidomic screen with Peter Michael. And what we identified was that certain lipids, in particular ceramides and sphingomyelins, were elevated. If they were elevated, had a really poor outcome, no matter what treatment you gave these men. And we're not talking a few months here and there, we're talking half the median survival. Those with the abnormal signature, which represents about a third of patients, had a median survival of 11 months versus 21 months. So this was a pretty impressive, we were pretty impressed by this. And we validated in an independent cohort, and we saw pretty much the same thing. So then what we said, well, how are we going to do things about this? Nobody in prostate cancer wants another prognostic marker. You know, you're sitting in front of a patient. No patient wants to be told, hey, you hit the jackpot. You've got the poor prognostic marker and there's nothing we can do about it. So prognostic markers in prostate cancer in the metastatic setting are fairly useless. So what we then said, well, what's actually happening? Why are the ceramides up? How can we potentially modulate the lipid levels? And we've started off doing something really simple, which is taking simvastatin, because statins are known to downregulate ceramides, and there's lots of studies showing that. And the first thing we're doing is just giving statins for 3 months to these patients, doing the lipid signature before and after to see if we are actually changing the lipid signature. It's a really simple, it's got a 1-page case reporting form. They take a blood test before and after, and simvastatin is about $1 a tablet or less. It's really, really cheap. And we got an ANZUP below the belt grant for just a small amount of money, for $30,000, that will allow us to run this because it's a very pragmatic study. However, if it gets the answer we think it's going to get us, that the ceramides go down, then we can start looking at the efficacy study. And in prostate cancer, the efficacy studies are happening. I think likely to be unsuccessful unless they look at the plasma lipidome, because it's the concept of, we talk about personalised precision medicine in every other part of cancer, but what about the body? The cancer doesn't live in a pocket. Lives in the body and it interacts with the body, and therefore metabolic interventions that affect the body that then affect the cancer probably should be just as targeted. So we've now incorporated this into the lipidomic signature simultaneously. It's being incorporated into a randomised trial out of the Gustave Roussy of using statins and aspirin to try and modulate metabolism. And my bet is it'll work for the third of people that have the poor prognostic signature. So there's many ways you can skin the rabbit in this scenario. So what have I learnt? I've learnt that really good science is essential, and it doesn't matter how many good connections you have. If you don't have good science and lots of good scientific collaborators, it's not going to work. I believe early clinical engagement is essential. Drag them in. It's really important, and sometimes they won't be in your institution and they won't be in your network. And what I'm really enjoying, having spent 20 years in oncology in Sydney, is that we are much more interconnected. There are a lot more clinical research networks that cross our institutions now and that we can take advantage of. Early pharma and biotech engagement, the same thing. And very much getting to know your legal and business development team. I have found that essential, as I have tried to work out what on earth the MTA and the this, that and the other means, and how badly am I going to be taken down if I want to publish. And patience. This work has taken, to get this into clinical trials, to have 2 out of 3 clinical trials currently open across Australia, has taken myself and our group 6 or 7 years. But I think it's really important to fight what you believe in, even if some days you feel that nobody is listening. We'll get there in the end. And this is the tip of the iceberg of all the people that have contributed to what I've just shown you, and I'm sure I have forgotten some people. But it takes not just a village, it takes a town.
Yeah, so, there, on the web, there's a protein atlas. So then, they have a certain, they have patient data where they can tell you, if your protein is highly expressed in this particular cancer, then they tell you the relative survival time. I'm just curious whether you have looked into that for your protein.
No, we haven't done that specifically. We've looked amongst our own cohorts, so for triple-negative breast cancer, for prostate cancer as well. But there's a lot of resources online for RNA as well as obviously for protein, and it's obviously a really good point to make, and I think that's key when you're trying to move forward. You need to have your target expressed, but also, as I sort of mentioned at the end, I think having biomarkers to tell you whether that protein is not just expressed but is actually relevant is also a really critical point.
I'd like to come in here too. So the target that we work on in Protein Atlas, that data is really dependent on the quality of the antibodies. And so we have data in our lab that show in cell lines that we're getting very different profiles with the antibodies that we have relative to what's in Protein Atlas. Now, I'm not saying we don't explain that, but it's very dependent on this.
So if the panel members can please introduce themselves before. Yeah.
Kieran Scott's my name. I'm from Western Sydney University.
I'm Peter Gunning from the University of New South Wales, but also from Trobio Therapeutics, which is a spin-off company we've formed.
Right. Edna Hardiman. I'm also from the University of New South Wales and Trobio Therapeutics. I'm Mamta Pora from the Health Science Alliance Biobank from the Translational Cancer Research Network based at UNSW.
So if I can ask a question. Lisa touched on the importance of precision medicine, and certainly the top tier pharma companies have active programs in precision medicine. And there is a strong movement towards co-development of drugs with the accompanying companion diagnostic. So is this something that you are considering very early on? I mean, certainly Lisa has touched on it, but for the other panel members and for the speakers, is this something that you are considering in your project planning?
Anyone want to take that?
Look, I think our experience is, do you actually have a visualised path to trial and market? Because in the end, if you haven't thought that through, and what that means is, what kind of patient cohort would you imagine taking your drugs into? What kinds of combination therapy would you imagine? What kind of biomarker data do you need? On the one hand, to know whether the patient is actually suitable, and on the other hand, any surrogate measures you have that allow you to be sure that your compound or drug is actually active in the patient who's being treated.
It's interesting, certainly from the clinical trials perspective, the drug companies play lip service to this actually, because what they really want to do is give the drug to everybody. And a biomarker in an industry context is not worth very much because the patient might only have it once, rather than the drug where they're going to have it multiple times. So in fact, certainly across the clinical oncology forum, what we are seeing is many more of the biomarkers coming in from academia. So academia is really, really important in this area Your drug might make it to market without your biomarker, but increasingly it's going to become important, and the company will be less interested than you will.
Thank you.
Yeah, I'd just like to add that I think this change really creates opportunities for people that we haven't really even thought about. You know, precision medicine, in my view, challenges the big pharma business model, and that then creates opportunities for others. So that's really the niche that Australia, in my view, has an opportunity to make a great impact in.
Just like to ask the panel, in the context of, I think, precision medicine today, we're getting pretty good at target identification in patients, making treatment recommendations, often for a single agent. Sometimes it does work, but most of the time it doesn't. So Lisa started to touch on this, is clever biologically driven combinations. What does the panel think about how quickly and how critically we have to move towards combinations, multiple targeted agents plus regular chemo backbones, this new age that we need to move into?
One point I want to make about that, and this was, I guess, being a basic biologist and being kind of thrown into this sort of translational context, for me, one of the biggest things I'd always had in my mind, you've got your target, you make a drug that hits that target, and then you test it in a clinical trial. But standard of care exists, and everything is within that context as far as, how you get things into clinical trials to start off with. So, I think that was a bit of a change of mindset for me, because initially you need to be thinking about combinations, or the population being quite a late stage population. So it changes what, as a biologist, you think it's a target and a drug and a cancer, and that's it. But everything is in the context of combinations, and I think partly that's needs to be set up in your preclinical models and your testing as being something you think about up front. So I think it really is key. And that's sort of why on my last slide I brought in the biomarkers as well, because I think if we can start to work out which patient populations to narrow it down into, hopefully we would then see less failed clinical trials by doing exactly what the pharmaceutical companies try and do, which is treat everyone and hope then to make more money.
I'll just make a couple of comments. I think the first issue is we need to broaden our what we call precision medicine, because precision medicine has largely been across the world the genome, which has been useful for arguably 30 to 40% of people. But I actually think that ultimately precision medicine, personalised medicine, will be a suite of biomarkers that includes genomic alterations, but there are other things that will be useful in that context. I think also that when we get to the combinations and how do we use these things, I think the biggest casualty we have of clinical trials is the science. Because once you get to a clinical trials management committee, almost nobody on the committee will be a scientist. And then there may be clinician scientists, but the person who developed the molecule and knows more about the biology than anyone else is generally not sitting on the clinical trials committee that then's running it. And what you really need is more scientists embedded in that phase, because when it doesn't work or something weird happens, or actually it really does work in a population you don't expect, you desperately need to go back to the science and understand it, because the combinations have to be scientifically and rationally done. And I hope I don't offend anybody in the audience when I say, if I have to sit on one more teleconference where, if it's not working, let's put it with a checkpoint inhibitor, here goes the pembrolizumab, I'm going to scream. That's the answer to everything at the moment. And that's why we need the scientists in the room, and I really am passionate about getting scientists onto our trial management committees to actually read us the riot act when we're going so far off the biology it's not funny.
I might just segue at this point, because we've got Mamta up here. So I think it'd be great to get an input about biobanks, and particularly, we're talking about biomarkers and how we can best access them, how we can best utilise them in terms of making sure we've got the samples from these clinical trials to be able to test when things go wrong and try and work out what's happening. So it'd be great to just get an idea of how that works.
Yes, the Health Science Alliance Biobank is based as one of the flagship programs of Translational Cancer Research Network. And the way we operate is we have an agile sort of model where we work with researchers to get the samples that are not within the biobank as well. So these are the boutique requests, and we have systems and structure and processes in place. And Caroline Ford is in the room as well, who's one of our valuable user of the biobank. So I guess I'll get her to comment on the ROR 1 and ROR 2 expression. But
thanks, Mamta. I guess the only thing to add is we work on gynecological cancers, which are quite rare, so we've found the HSA biobank incredibly useful in getting access to some of those tissues. But also, they've worked very well with us with going back and getting control slides and recurrent metastatic sites from samples as well. So it sort of goes beyond just a single TMA or something like that. You can get a real wealth of data from some of these biobanks. The question Jeff was asking more about clinical trials, I think is also really important. So we're now soon going to launch our first clinical trial, and that's very much a result of the target identification which has been facilitated by the biobank. And so the processing of those samples and how we will actually conduct the translational research that's part of that clinical trial is very informed by the scientists, not the medoncs. So working on what Lisa said as well, I completely agree with those points. Happy to chat further in breaks with people.
So, I mean, just to sort of add to that, I mean, from what I presented, you know, we did a lot of TMAs from various different biobanks to look at our marker, and I think it's a really critical point. And things like the HSA biobank, as Mamta said, you know, when we've gone to them, you know, I've only been at UNSW 15 months, and sort of said, oh, we're looking to get some prostate cancer samples and try and work that up. They're able to help us try and facilitate that, and in terms of actually collecting, you know, different samples at different time points. And so, you know, it's good to know that these sort of resources are out there because, you know, again, you know, from where I was 3 years ago, pigeonholed in a basic research setting, to know that all these resources are there where you can actually really accelerate your translational research by utilising really precious samples that have been collected and annotated really well. I think it's really key. I would encourage people to go and talk to the various different biobanks and see how they can help you to actually do that.
Just continuing with the personalised medicine theme. Lisa, you really nicely showed some of that explant data. I'm just wondering what you think about using that model to guide a personalised medicine program.
I wrote an NH&MRC grant that got trashed about that. I think it's look. I'm really excited at the thought that we could do that because it's essentially you could turn around data in 2 weeks, and the answer is yes. I think it should happen. I actually termed it human avatars. I was hoping for a little bit of, you know, imagination. The NH&MRC didn't like it. But we'll get there eventually. And I think it could be used, this same assay has been used in breast cancer. We've now got some follow-up with Lisa Butler on some of the earlier samples that she did, and we're starting to analyse those outcomes. But the key way to do it is in a clinical trial and start with your standard of care drugs and prove that it can predict response to your standard of care drugs, and then start using it to do more exciting things out the other end. So anybody who wants to revisit it, I've got a really nice written grant and I just need to find the right slot for it. Happy to collaborate.
Thank you to all the speakers. Jeff, Peter and Edna, you've gone down the brave path of, you know, the startup company type venture. I just wondered, as academics, what do you think are the biggest challenges and what gaps do we need to fill to support people like you in academia?
I'll address that. So as Phoebe mentioned, I'm an academic, and the opportunity presented itself for us to start up a company. So we've been pursuing a family of proteins for 30 years. So there was a lot of science behind what we had. We knew that it was druggable. We had demonstrated that. And we then had the opportunity to take a CRCP
fund, which was quite substantial, but we had to form the company to support that. The biggest challenge for us has been support, infrastructure. We have to come up with money in order to set up our company. And you have to have that money before you can even then go to venture capital, do that appeal. And then you have to be able to know who to go to. It's that basic support that I found lacking. So we had to rely on personal networks. And now I've had the opportunity to see what is available. Say, we have a colleague we're forming a joint venture with in the UK at Cambridge. She's in the Abraham Accelerator. What is available to her is absolutely phenomenal in terms of monetary support. She has venture capital coming to her every other day saying, show us what you have, we may be interested. And now I'm also aware of Stanford Spark, who has an outreach program, who has these extensive networks that we may be able to plug into, and other outward-looking programs in other countries. But I haven't found it here in Australia yet. So those are the programs that I'm going to be plugging into, but it's a hard slog if you have to go it alone.
So in our last session, we have the director of Spark Oceania, which is Professor Michael Wallach, who's really supporting commercialisation and technology in Australia and in the Asia Pacific. So we have that opportunity to speak with Michael.
I was just going to add to that. I mean, I guess when I started my company, I was at Centenary Institute, so we had access to the Medical Research Commercialisation Fund. So for us, it was a little bit easier because we kind of skipped in the middle part, and they came on board and told me everything I needed to know. So, for me, it was quite a different experience because, they put in place all of the structures around, bridging all of my lack of knowledge in the area and really helping us push forward. So, obviously we have representatives from CCI and excuse me, and Garvin, and I'm not sure if Ingham as well, but the institutes have access to the MRCF, and it's really quite a straightforward process with the pre-application and being able to move forward. And it was substantial funding that we got, millions of dollars to actually move that forward. So I think it's knowing what you have access to. Uniseed, who came on board, have much less funds available, so it's really sort of the hundreds of thousands, if that. So it's much harder to kind of use that funding to really do something substantial in that space. So, I think it is trying to see the opportunities where they are, and obviously if you're in an institute, there is a lot of money available through MRCF if you tick some boxes in terms of what they're specifically interested in.
So in going down the startup pathway, what made you choose that pathway and not simply license out to a pharmaceutical company?
Me again. I was going to have some water. Yeah. So I've been talking too much this morning. So
what was part of your decision-making process there?
I think our main decision-making process was we had a very early hit compound. We had nothing that was a pharmaceutical company would remotely be interested in. So normally MRCF wouldn't touch what we had, but they really liked the target. And so that's why I sort of focused up front on the target, making sure, I think that, the cancer cell paper was critical. Someone else's work showing it's a good target as well. Our work had shown the target was there, that we could knock it down in vivo models. So we had the biology kind of set, and we had a hit. And despite that hit being, not fantastic, they were willing to take the risk. We were nowhere near at a point where a pharmaceutical company would be interested in that. But through the process, we had a number of pharmaceutical companies that were interested, but we just never got to the point of having a good enough molecule where they would make that jump. So, I think you need to know where you're at, and, different scenarios will be more attractive to different circumstances. So yeah, I think for us, we were just fortunate we sort of fell into the right opportunity and being in the right place at the right time.
Peter, can you comment on this, please?
So my sense is that the pharmaceutical industry is interested in de-risking to the extent that show me you've actually got something useful that has advanced through phase 1 is attractive. Because if you look at the graph of how many molecules do you start with and how many come out the end as a commercial product, what you're looking at is a pathway where there is an enormous attrition of molecules. So in the conversations I've had with Merck and AbbVie, the conversations have usually come around to show me something after phase 1.
Yes, so how can we in academia de-risk ourselves? Because we're hearing the same thing over and over again. And I think the licensing route certainly, from my perspective, from what I'm doing, is a preferred route. It will get the technology out there faster. Regulatory requirements will be supported by the industry. So how can we go about de-risking ourselves?
Look, I think part of it comes down to we just have to get more clever at initial phase 1A at least trial. I think in the end, having molecules developed that go in, and look, just so we're clear, I'm just talking about academics developing completely new drugs and then getting them to a point where pharma is interested. And I think in the end, if you don't get your molecules to a point where you can take them into, at least initially, some patients looking at phase 1A, I think it's really difficult. It's that relationship with clinical colleagues who are sufficiently convinced that there is potential value in your molecules and their target to collaborate with you to start to take it into clinical trials.
Yeah, I'd certainly agree with that. If you can get a clinician on side, then that's, in my view, 90% of the thing, and that's certainly been my experience with our early phase clinical trial that just finished last year. With this was driven by a clinician who saw the data and thought this trial is worth doing. So it's a very strong view there.
Lisa.
And in order to find the clinician, if you don't know one, just email the head of department of one of the big units. If you know a unit sitting next to you, just email. We used to get in cold calls. You know, it doesn't require an established relationship. Medical oncologists, and this spans beyond cancer, clinicians love new things and they'll really enjoy having a conversation with you. So just email them.
Absolutely. I mean, that's how everything worked with us, transitioning from prostate cancer, where we knew clinicians, to breast cancer, was just Jane Beath, she's at RPA, that's next door, go and have a conversation. She was just thrilled with the idea of coming on board with accelerating something through to the clinic. I mean, that's what clinicians are trying to do. They love to have new opportunities for their patients. So it is. I found it surprisingly easy to get clinicians to really partner with you and come on board. So yeah.
I think there's a question here. Yep.
Yeah, we've heard two of the panel members talk about interactions with VCs. Jeff spoke about things that he would do more of in the future, i.e. describing the risks associated with the programme. I wondered if there are some things that you would perhaps do less of Are there any mistakes that you made in those initial interactions?
I think the entire initial interaction was mistakes from my part. As I said, it was an extremely positive experience because I really knew nothing about the process. And so they just kept telling me the things that I needed to do. And so I learnt a lot by the fact that I didn't know a lot. So looking back, what would I do differently? I think I would follow their advice. I mean, I think that's what we did, but I think it's thinking about the target market and putting that as sort of the number one. How are we going to get this into the clinic, and then work backwards? And so, everything that I had done was thinking about my research, and I've done this and I've done that, but it just doesn't become about that anymore. It's, you work backwards. Okay, so you've got a target market, triple negative breast cancer. If we want to get into the clinic, we need clinicians, clinical trials, and then you work your way back. Okay, well, in the end, you get back to medicinal chemistry, and you need a molecule, and you need to make that better, and need to get into preclinical models. But it's that really sort of changing, rather than what's the next step forward, and go to the end and work your way back, and that's then the pathway you need to take to get there. So it sort of reversed my, okay, I've got this, now what do I do next, to thinking about the future and then working backwards.
One last question.
Yeah, quick one for Francis. So if I understand you correctly, the target of your compound is still not found yet, right? So my question is, will that in the future have a negative impact on the development of this therapy?
I guess that's something that I need to find out as well. We know that some of the biggest drugs in the market, such as metformin, they often don't know the exact way these are working. So that's what I need to find out, is if I do this phenotypic screen, how important is it for me to identify a target? Is it critical for me to move forward? And maybe people in the audience or on the panel might be able to give me more insight into that.
So I'd like to thank all speakers and the panel members for a very engaging and active discussion. So please join me in thanking them. So Greg's talk is high throughput assays and drug screening. So please join me in welcoming Greg.
What I'm going to do is just take you through a little bit about assay development in the context of doing high throughput small molecule screening. So first of all, just cover a bit of the drug discovery process. We've touched on this today a little bit. Tell you what we describe as high throughput screening, some of the considerations that you need to take into account in order to do that screening and get out the best chemical hit you can. And I'd like to finish off the talk talking about three things or entities that you might not know about that we have access to do high throughput screening and small molecule drug discovery through or at UNSW. So if we look at developing a new drug, obviously it's a long-term process, years and years and years, as we've heard, and many hurdles to go through. And basically the process starts off at target discovery and makes its way through the various stages of clinical trials, regulatory approval, etc. And so when you're talking about target discovery, and we heard a little bit about that this morning, you want to make sure that your target is relevant, that it's linked to a biological process or a phenotype related to the disease that you're actually looking into. When you get to hit identification, then you're really looking at trying to develop a relevant biological assay that can be robust, that can then be used in a high throughput screen. A high throughput screen would be using large chemical compound libraries, screening against either a target or the cellular pathway itself. And of course, taking this kind of approach is a high risk because, of course, you have very few of your compounds that actually qualify as hits in themselves. And then once you have a chemical hit or a chemical hit series or more than one, you want to go through the process of optimizing those chemical hits and making them more drug-like. So you want to improve selectivity, safety, etc. You're synthesizing or having to synthesize a wide variety of different chemicals. And this stage really does require a considerable amount of medicinal chemistry manpower, and I'm hoping that that'll be touched on in the talks coming up after mine. And then finally, you have the preclinical work, developing your preclinical package, and we've heard a lot about that already this morning. You want to be looking at the physiochemical properties of your drug, so how is it metabolized, how long does it exert its effect, what are the potential side effects, and so some of these features need to be taken into account to have that preclinical package. So what is high-throughput screening? Well, it's one of the main approaches that has been used to try and identify a starting point. That is a chemical series itself. There are other technologies I don't have time to touch on today that you can use either in parallel with HTS or leaving HTS behind. Now, the actual process itself probably can be broken down into 3 different stages, and those are assay development, primary screening, and secondary screening, and those are defined there for you to look at as is. But the whole process of high-throughput screening is a multidisciplinary undertaking. And so you have to have a lot of factors come together, and that is you have to have good assay development, you have to have high throughput or high content screening expertise, automation systems and robotics, a way of managing compounds. So we have access to a 300,000 compound library. How do you manage that itself? And then data analysis, management of that data, and informatics to look through that data and actually call a hit a hit. So the key to all of this is your assay. And so we kind of stand by the rule of 3 R's, which actually turns into 4 R's, as you'll see here: relevance, robustness, reliability, and reproducibility. Secondly, is the assay that you're doing too technically complex? You've got to keep it quite simple, even though you may be dealing with a complex biological system in the form of cell-based assays, for example. Your assay has to be affordable. You can't be going in and having extremely costly reagents and screening 300,000 compounds. And finally, you want to be able to have your assay miniaturized so it can be automated. So the take-home message from this is the quality of an assay determines the quality of the data. Garbage in, garbage out. What kinds of assay strategies are there? Basically, you can break them into 2, phenotypic and targeted. The phenotypic assay is usually a situation, as we heard from Francis today, where your target is usually unknown and can stay unknown for a longer period of time than you'd like. But the readouts are generally more physiologically relevant. But as we touched on, you need to have a good target deconvolution process after that. The other area is the target-based approach. So here's where you have your known target, you're actually then screening against the activity, and you have a readout for that. But again, you're dealing with an isolated target, and it may not be physiological. If we then go and drill down further, from a technical point of view, you're really dealing with biochemical or cell-based assays. And I won't go into all the minutiae with each of these areas, but more cover the advantages and disadvantages. So with a biochemical assay, where you're really looking at screening against a target, it can be simple and make the assay consistent and reproducible, and you have direct contact and measure of target engagement. However, you have this battle with whether it's physiologically relevant, and you can't really deal with a lot of the possible off-target effects of your compound. Cell-based assays, on the other hand, you're looking then at screening on the function of your target in the cell context. So you're looking at possibly a more relevant functional outcome with your compounds. But again, high-throughput screening can be quite complex. It can be simple in one sense, from cell death screening, or it could be highly complex with high-content multi-parametric screening. So what are the kinds of assay readouts that you can use? And I use the word some because this is a very, very short list of all that you possibly could use. And I won't touch on all of these today, but simply to point out the more common ones around readouts of absorbance, fluorescence, luminescence, and then more recently, if we go down to the bottom, high content imaging, where you're looking at multiple parameters in your readouts. What do you need to do to optimize your assay and validate your assay? Well, there's a lot of factors that you need to consider. Some of them here: protein concentration, substrate concentration. If you're doing cell assays, cell density, reagent concentrations, your compound exposure, and the timing that you have that compound exposed. Probably one of the major critical things, though, is your controls, because you're going to use your controls to define actually identifying your hit. So you have negative controls; those are fairly straightforward, but your positive controls can be two forms. They can actually be assay controls, or they can be biological controls. Once you have an optimized assay, you've got to validate that assay. And what I mean by that is you have to have an assay where if you screen a single compound in a single well one time at one concentration, you can be confident that you can call that a hit. All right? And so a validated assay to us in an HTS facility has to be reproducible from well to well, from plate to plate, and day to day. And of course, we use a variety of statistical parameters or analyses to actually define whether an assay is of the quality for high throughput screening, and I won't go through these in detail, but one of the key factors is the Z prime factor, which provides us with basically a measure of the readiness of your assay for HTS. So what I want to do in the last couple of minutes is spend a bit of time telling you a little bit about some of the, centers that you can actually access as UNSW researchers trying to start out in drug discovery. One of them is, in case you don't know, your campus has a ACRF Drug Discovery Center. So we have the infrastructure and we have the expertise to do small molecule screening and follow-on work from that. And we have for years provided a fee-for-service drug screening service. So we're based at the Children's Cancer Institute, and that's in the Lowy Cancer Research Centre. And of course, we're a member and partner with a variety of the Australian drug discovery and development stakeholders and consortia. So, we've been in business for 10 years. We've done over 100 drug screens, and they've included target, cell-based screens, and high-content screening. And so we've done everything from assay development, adapting assays to automation, miniaturization, HTS, HCS, generation of chemical hits, and worked with companies on their analog screening as well as part of their lead generation and lead op programs. And so from that, we've discovered novel chemical matter, we've repurposed drugs, found unique drug combinations, and also done drug sensitivity testing as part of a personalized medicine program. However, we're going to close that down, all right? And we're going to come up with a new operational model, which I want to tell you for the first time, and this is the first time we're telling you about this. This is going to be an end-to-end pipeline for drug discovery, where we'll be going from target to candidate selection and developing key preclinical packages. We're going to have access to world-leading drug discovery researchers who have been part of our scientific advisory committee for years, as well as technology platforms needed and the expertise along that pipeline. And the exit strategies for this new entity are going to be commercialization and finding ways to do that, and as was touched on the panel here, getting things into early phase clinical trials. I can say up front that it's going to be an extremely rigorous process that we're going to put in place to select projects as part of this therapeutic innovation group or center. What will that look like in terms of the drug discovery continuum? Well, we're going to fit in really in the middle here. I've kind of pointed out in blue where we're going to have our attention. So targets will come into the therapeutic innovation group from a variety of sources: academic researchers, commercial partners, companies, and some of our own in-house partnerships. And then out the other end, hopefully, will be candidates that can be nominated, licensed for further development, and or enter into clinical trial. So where's the value add for the Therapeutic Innovation Group itself? Well, we have access, as I said, to a number of Australian and New Zealand leading drug discovery researchers, and what I mean by this is the people that have taken them to commercialization and or the clinic and or have drugs on the market themselves, and they'll be advising us. We obviously have the assay development, the HTS, the HCS capabilities all on campus right now. We're going to get access to medicinal chemistry, protein production, structural biology, the ADME talks, PK/PD that needs to be done, and the relevant biological models for the particular targets we take on board. So I'm telling you about this today with the hope that a number of you will come and start discussing things with us as to how we can move forward and partner with you. However, I've been asked that if you want to now do fee-for-service screening and you have a good assay, that is an assay in a 96-well format, may I introduce you to the National Drug Discovery Centre. This is based at the Walter Eliza Hall Institute in Melbourne, and what they offer is basically 90% subsidized, government-subsidized screening access. You get automated robotics, diverse readouts, and they have 300,000, the same 300,000 compound library that we have access to, but they can provide screens at significantly lower costs. And there's what you need to do to apply. You have to have a working assay in place. You can be basically from a university, a research institute, or a company. There's information on a couple of the links there, and their first round of applications are closing on the 7th of November. So I will end there. Thank you for your attention, and just remind you of the Therapeutic Innovation Group that we're developing, and we would love to talk to a lot of you about it at morning tea or during the panel discussions. Thank you.
Our next speaker, Dr. Eleanor Eiffe, High Throughput Assays and Drug Screening.
So I'm going to talk today, I think my talk actually is a really good follow-on from Greg's presentation then, because I'm talking about the next step of drug screening. So how to set up a screening program to take you from that initial hit into a compound that's good enough to go into the clinic. So it's all about getting the right sort of information at the right time. And I've subtitled my talk Fail Fast. So that's an idea that comes out of startup culture, and I think it really is applicable to drug development, particularly when you're working out of an academic group or in a small startup. You don't have a lot of resources, so this idea of failing fast, it's often said fail fast, fail cheap, is a philosophy that you can apply to your screening pipeline. So as I said, I'm talking about that the screening that happens sort of after you've found your hit. So you've got a validated target, you've found an initial chemical hit, and now you're taking that first compound that's shown some activity, doing some medicinal chemistry and transforming that into the best compound that you can take into the clinic. And it's really about, you need to set up your screening program to make sure that you are finding that best compound. I'm talking also about screening in terms of feeding the design cycle for medicinal chemistry. So I'm actually a chemist myself. So this medicinal chemistry, as we've seen in a few of the other talks already, it's a cyclic process. So you design some molecules, synthesize them, and then screen them, and then look at structure-activity relationships, structure-property relationships, and use that to design the next set of molecules that you're cycling through. So really, I'm focusing on the screening that feeds into that cycle and how you can make sure you're giving your medicinal chemistry collaborators the right information so that you can make the right decisions about which molecules to take forward. So a little bit of background on the project that I've been working on for the past 6 or so years now. So this will just give some context for some of the examples I might talk about later. So I'm working with the Cellular Genetic Medicine Unit at School of Medical Sciences, UNSW, working with Peter Gunning and Edna Hardeman on a protein called tropomyosin. And we're specifically looking at an isoform of tropomyosin that predominates in cancer cells. So tropomyosin, it's a coiled coil. It polymerizes along actin filaments in both the cytoskeleton and also in the muscles. And there are over 40 different isoforms of tropomyosin, and each isoform gives the filaments a different effect. And as I've said, we've noticed that in cancer cells, and this is pretty much universal across a lot of cancer types, they rely very heavily on isoform TPM 3.1. So if we can come up with small molecules that disrupt the function of that particular isoform, you have a way of selectively killing cancer cells while leaving normal cells basically alone. So the idea is that we have a small molecule, it binds to the coiled coil and then gets incorporated into the polymer around the actin filament, and that tropomyosin with the small molecule in there can't work properly. Essentially, it can't regulate which proteins bind to actin, and if you're inhibiting 3.1 in a cancer cell, that leads to cell death. So I know we've seen a lot already about the overall drug development process, and it's something often described as like a funnel. The idea that you have potentially thousands of molecules which eventually gets whittled down to a very small number that actually make it to market. And the thing is, as you progress along this process, not only the number of molecules are decreasing, but your costs for each stage of the process go up. So at the initial stage, when you're looking at sort of lead identification, lead optimization, you have a lot of molecules, you're making a small amount of each of them, you're doing a lot of in vitro screening. Then as you start to find some better molecules, then you go into animal experiments, start looking at formulation, that requires more compound. Then when you've got your candidate that you're looking to take through to the clinic, you have to meet all the regulatory requirements. So that's looking at studies that have to be performed to GLP, or Good Laboratory Practice Standard, which means you need higher quality compound. You also at that stage need to start manufacturing this on the hundreds of gram scales, because you're going to start doing long-term toxicity studies. So that by the time you get to the clinic, then you've committed to one compound, and you've committed to manufacturing that to a grade that can go into humans. So you can see as this process goes along, it gets more and more expensive. So you really want to make sure that the compound that you're taking into the clinic is the best compound you could be taking through. So it comes back to that idea of failing fast again. You want to find the wrong compounds early. You want to be ruling out anything with an unacceptable risk, either that it won't work or that it's going to be toxic. You want to find that out when you're still in the early stages. Because if you take the wrong compound into the clinic, you're talking hundreds of thousands, potentially up into the millions of dollars that you've spent on the wrong compound. So just to go really, really broad for a second, so obviously the goal of drug development is to develop a drug. And what makes a good drug? We're trying to cure a disease. You also need to consider, though, sort of some of the practical aspects of what makes a good drug. So think, is this something that patients are going to be, want to take and be compliant with? So there's always the goal of a once-a-day oral drug is sort of held up as something that's, achievable, taking a pill once a day. So you need to think about that right from the start. If you need to make a molecule that is orally bioavailable and is going to be delivered at a reasonable dose. You obviously don't want to be taking grams of something to treat your illness. So you also, a good drug should be well tolerated, have minimal side effects. So it's all about, balancing these sort of benefits with the risk. And again, sort of that dose route administration can come into this as well, because you want to have a good therapeutic window. So your dose is very low, and your risk of toxicity doesn't come in till much, much higher. Also, you need to consider commercial considerations if you're looking to develop this yourself or a partner with a pharmaceutical company. You need to consider, is there a market for the drug? Also, do you have a patent position? What's your manufacturing process going to look like, based on the properties of the molecule as well? So there's a lot that you're trying to optimize when you're going from that hit to that lead to that clinical candidate. And you really need to be asking the right sort of questions from the beginning. And I'd like you to think about sort of setting up your drug screening program as looking for reasons to reject each molecule. I think one of the most dangerous things you can do is to have a favorite and just try and get your favorite along that pipeline as quickly as possible. I think you need to be really critical of every molecule that's coming through and really interrogate, does that molecule work? Is it going to treat your disease? And is it going to be safe? And really ask the tough questions of your molecules early on. So the first part of drug screening is obviously, does it work? You're looking for biological activity. And I think Greg went through really well sort of the difference between biochemical assays and cell assays. And also we've heard a little bit about looking at just binding assays where you have a target versus functional assays and also phenotypic assays. And I think all of these different kinds of assays all have a role to play because you're asking that question from different angles. So if you have a target, you can show that does your drug bind to the target? That's important. But then does your drug binding to the target have the effect that you want it to have? So in our case, we can look at whether our drug binding to tropomyosin inhibits the ability of tropomyosin to protect the actin filament. So that's that middle diagram there. So we can actually look biochemically, and we can also see this in cells. So we have some cell lines that express fluorescently tagged tropomyosin. So we can actually look at the filaments containing our target isoform and see, are they intact or are they being degraded, which is what we want. So it's also important to look at, I think, a combination of biochemical assays and cell assays, because something like a binding assay where it's just your drug and your target, that gives really useful information back to your medicinal chemist, because especially if they're doing structure-based drug design, because then you can really tie back the results of that binding assay to how well does this molecule fit in the binding site. But I think it's important to also look at cell assays, because a molecule that fits perfectly into the binding site of your target may not be able to get through the cell membrane, or it may also bind to something else that's in the cell. So I think it's important to look at the isolated system, yes, but also look at something a little bit more complex, like the cell assay. So in terms of molecules getting into cells, that also brings me on to the next part of the does it work question. Because I think that's something that is easy to miss, especially in the early stages of drug discovery. And we actually had a good example before from Francis in the first session, that pharmacokinetics is really important. So the efficacy of your compound in vivo depends not just on how potent it is, not how well it binds to the target, but also exposure. The compound has to get to your target. So that you need to consider what's going to happen if you inject it or if you feed it to an animal. How's it going to get through the bloodstream? Is it going to get heavily metabolized by the liver? Is it going to be able to get across membranes to get where it needs to go? And one approach that we've taken is to look at this really early. And not necessarily going straight to an animal PK model, but we've started looking at in vitro assays to look at these ADME properties. We've been doing this through a CRO, so we've taken, I'd say 10 of promising looking molecules sort of in the middle of that lead optimization cycle and said, all right, let's look at their solubility, their stability, plasma protein binding, looking at some membrane permeability assays, and also looking at their metabolic stability, which you can do in either liver microsomes, S9 fraction, or actually the full hepatocytes. So what we want to do with these assays is really looking for red flags for problems that are going to be with metabolism. So then we can sort of steer the medicinal chemistry program towards the compounds that work and away from the compounds that don't work so well. So there's something that comes up that Big Pharma talk a lot about when they're talking about drug discovery, and that's multi-parameter optimization. So not just optimizing for potency in your binding assay or in your functional assay, but in parallel with that, optimizing for properties. So it's all about sort of giving your medicinal chemist the right information so that they're optimizing for the right things. And you really need to think about the whole drug in that sense. It's also interesting, you can see sometimes in published work from pharma where they publish on a program that got to the clinic or to commercialization, and the compound that actually made it through is not necessarily the most potent one. And quite often it isn't, because it's not just about the potency, but it's also how is it going to behave once it's in the body. So the last thing I wanted to talk about is obviously one of the biggest risks that can come into a drug discovery program, and that's toxicity. And I think you need to be thinking about toxicity really early on. Because if there's something in, say, the chemical scaffold that you're working with, something that's inherent to your whole series of molecules, you need to find that out early so your medicinal chemist can fix it, or you can start looking at a different compound series to take forward. So one of the big toxicity risks, one of the most famous anti-targets that's screened against is the hERG ion channel, which causes, if you block that channel, you cause fatal arrhythmias. And this is actually one of the studies that you need to do for a regulatory submission. So for the regulatory submission, you need to do a full GLP patch clamp study, but you can also, earlier in the process, you can look at just a hERG binding assay. Just again, it's looking for those sort of red flags. Are any of our compound series likely to block the hERG ion channel? And that's something you want to know early so that you can take your medicinal chemistry in a more productive direction. So other risks you can look at for toxicity is looking at cytotoxicity, so for example normal cells versus cancer cells. And you might also, especially in the cancer space, need to think about the potential for drug-drug interactions, because there's a good chance in your phase 1 trials your drug is going to be administered in combination with something. So one of the things you can look at early on is just look at whether your compound inhibits the cytochrome P450 enzymes or induces them. And that's a relatively inexpensive assay that you can do early on as part of your selection process and also to feed back into the medchem. Something that also came up in Jeff's talk at the beginning is thinking about the questions that either an investor or an industry partner or even a regulator looking to approve your drug for clinical trial might ask, and really understanding the risks specific to your project. So these could be including, say, on-target toxicity. Is there something, will something bad happen if you inhibit your target too much? And that's all that establishing therapeutic window. And you need to show that, I think, that you've understood that and you've looked into it. And also then there's related targets, looking at your specificity, what potential closely related targets are going to have off-target tox. So for us, looking at tropomyosin 3.1, obviously we want to target this cancer cell cytoskeleton. We don't want to target the tropomyosin in the muscles. So that's something that we need to address specifically, and we need to be able to show to anyone looking at the project, have we answered that question? So just to finish off with some final thoughts, I think it's important in a drug discovery process to begin with the end in mind. Think about what makes a good drug, what's something that's going to help patients, what patients are going to want to take. And also that idea, think about, it's the idea of funneling. There are a lot of compounds that are rejected, and you want to make sure you're rejecting the right compounds and you're doing that early. Also, I think it's important that your screening is really giving the right sort of feedback to your medicinal chemists. So making sure that you're measuring the right things so that your medicinal chemists are optimizing for the right properties. And final point, again, comes back to failing fast. You want to identify the risks in your project early and anticipate those tough questions that you might get from a potential partner. Thank you. And acknowledgements, the Cellular and Genetic Medicine Unit at UNSW, and this work is also part of Therapeutics, and we're funded by a CICP grant from the government.
Our next speaker is Professor Jonathon Morris, and he'll be speaking about medicinal chemistry.
Thanks very much. Today what I thought I'd tell you about is, some work we've done in the group on developing what was originally a chemical probe that's turned into a preclinical candidate. So I'm a synthetic organic chemist sort of by training, and I've always been interested in natural products, working on biological systems, and that led us into some interesting kinase inhibitors, and then from there we've ended up in this project. So to give you the background of the project, alternate splicing is obviously a really fundamentally important thing in generating protein isoforms, and I pick out here VEGF as an example of how isoforms can help, and in this case VEGF actually has two different isoforms, VEGF165A, which promotes angiogenesis, but there's also VEGF165B, which is actually anti-angiogenic. And so my collaborator David Bates had discovered VEGF165B was obviously quite an interesting protein that would be of interest in controlling angiogenesis. Now, alternate splicing obviously relies on RNA going to pre-mRNA, and then it's the splicing together of the exons. Now, this is controlled by splicing factors, and clearly it's controlled by the phosphorylation of splicing factors that takes that splicing factor into the nucleus. And so, for instance, in this case, the kinase that's of interest is SRPK1, And it's a kinase that obviously phosphorates this splicing factor and then triggers the angiogenic form of VEGF. And so therefore the hypothesis was that if you could inhibit that kinase, you would be able to turn off that splicing event, and so therefore 165B would be produced. And clearly your body uses this splicing mechanism to modulate the protein. So that's sort of the idea. How the story started though was in 2011 I went to a conference in France. I was the only chemist in a big pile of biologists, and I got talking to a person called Stefan Knapp, who is actually one of the great protein kinase X-ray crystallographers. And he sort of established that I like to make compounds, and he showed me this molecule Sphinx, which is just a name for SR, protein phosphorylation inhibitor number 10. And he had this compound that he'd done a screen on for splicing kinases, and had discovered that Sphinx was actually quite good at that. He had a collaboration with David Bates, who I'd never met, and David had shown that you could actually obviously inhibit angiogenesis. The other thing that Stefan was really great about was that he actually gave me the coordinate. He gave me an X-ray coordinates as well, so he actually had a crystal structure of this compound bound into the active site, or near the active site. And so I did ask him at the time, I said, surely someone at Oxford might want to make some of these molecules. And they said, well, yeah, no one was interested. Too simple a molecule. So an interesting insight into medicinal chemistry sometimes, of complexity for academic chemists is often something that we really like. But this molecule is obviously small. It's got a few interesting things in it. It's got a CF3 group. It's got an amide, which is always going to trigger concern in medicinal chemists' minds. But I guess, a bit like Jeff, I didn't know much about stuff, so I decided to get going, and I got an undergraduate honours student to actually make 10 compounds. And she was able to make a compound that was reasonably active, and the second honours student then made another batch, and that led us ultimately to this molecule, Sphinx 31. So 31 compounds made in a couple of years with no funding. This was just a sort of a fun little project. And you can see, if you look at the molecule, what we've done there is we've actually varied we changed the methyl group to a 4-pyridine, and we've changed the saturated heterocycle there to a different type of one. And so what we were really doing was just using the X-ray coordinates and trying to find different parts of the molecule that would interact with the kinase. And there's a lot more to that, of course, that we could talk about, but I don't think you nearly want to hear about all the synthetic parts. But we did establish that the CF3 group was really important. The amide was also very important. In actual fact, it's part of the binding mode. And what we found is, and the part of the story I didn't also say, is that I still haven't met David Bates. I didn't even know he actually had my compounds for the first 6 months, so Stefan had actually just given them to him, and so he wrote to me and said, I'm filing a patent, would you like to be on that patent? And he was at Bristol at the time, and I said, sure, no problems. He said 25% enough, and fine. Then, of course, he then moved to Nottingham, and when he moved to Nottingham, they were looking to spin out companies, and so he was basically said, we're going to spin out a company, and we filed a second patent, which is covering Sphinx 31 there. What he had found, of course, with this work was that, and so with this angiogenesis, there's a number of things you could do. You can do cancer, but also you can do pain, and there's a few other things. But the one thing that we're really focusing on was age macular degeneration. The fact that controlling blood vessel growth in the eye is a really interesting thing, and so you can also, a very easy experiment to do where they can do the laser etching of the eye, and then you can use the inhibitor to see whether you're actually getting blood vessel growth retarded or not. And so this is all the data going through here. What it establishes is that compound was non-cytotoxic, it was in the eye, it was doing things that we wanted to do, and this sort of triggered this idea to spin out a company. And so Xnate was bought. Now, interestingly, I wasn't a founder of that company, and that's sort of because it was created in England through Nottingham, and as Stephen Palmer and I will discuss later, maybe it certainly creates some interesting challenges when you're moving forward. But Exonate was born, and originally it was going to work on cancer, pain and eye disease, but very quickly it became a project about eye disease. One, because we could see the market. We also were able to sell that story much more easily to some investors, including Uniseed, and so therefore our work was sort of focused on that. So basically I became head of medicinal chemistry for Exonate, working sort of at night while everyone else was in England. So worked out quite well. And we had a pretty good system in terms of they gave me money to do some design and development. So basically we were building ideas of how to make the molecules, and then eventually we would then send them the chemistry to India, and we'll have a CRO there generating numbers. What I forgot to mention though is that this is so the challenge here was potency was never a problem. In fact, we got to the point where the biologists were complaining that I'd only sent them compounds with 10 nanomolar inhibitors because they were just saying it's not potent enough. But we were very easily able to get potency, but what we really worked out was we need to understand how to get the compound into the eye. So, and essentially, we had a medicinal chemistry consultant who has been really valuable to me. He was our second one. And basically what we found was that we could do this, and what we really were doing is all the safety stuff and all getting penetration into the eye was actually the big problem and the big challenge. And every time we solved that problem, we'd lose potency. So it was that usual thing of going back and forth. We made 600 compounds in total in the last 5 or 6 years. And part of that was because we were tiny, not a lot of budget. And so therefore we had to be really sensible about what we could or couldn't do. We also did generate some alternate scaffolds for backup series, and a series of patents were filed. So to cut a long story short, we now have a molecule that is being developed to go for clinical trials. The big thing for us, we got Wellcome Trust funding about 2 years into the project, about £5 million to actually generate a preclinical candidate, and from that we're able to do this. And so what you can see here in these slides is essentially 3 compounds where we're doing a sort of like a heat map of the eye, where you're obviously using mass spec to track how much of the compound is present. You can see compound C clearly getting high concentrations early, and it persists as you go through. And the third line there is showing you that it's 14 hours after the final dose of 6 days of dosing. So these molecules are going into the eye, into the retina, staying there and suppressing blood vessel growth and looking like a very good treatment for eye disease. So basically we have compounds that inhibit as an eye drop, and I forgot to mention that the current gold standard for AMD is a hypodermic needle injection of VEGF antibodies to the back of the eye, which should be done every 2 or 3 months, quite expensive. So the goal here was to have an eye drop that we could actually deliver every day and at concentrations that were reasonable. And essentially we've achieved that. And essentially that molecule is now going forward into preclinical trials. Pharma has now found us and decided they would really like to invest or purchase, and so that's currently what Exonate is dealing with. And so it's a nice little story in terms of it started off as a really molecule that was a probe. It wasn't very soluble, it wasn't very particularly active, and we were able to then play around with that to get a compound that's potent. Solubility is still a bit of a challenge, but it's good enough. Interestingly, the amide is actually really good for us because it actually falls apart once it comes out of the eye, which means that we don't have other effects. So in actual fact, it's been a useful thing for us. What I thought I would like to do, though, is give you a couple of sort of take-home messages and lessons learned as a chemist. It was one that you don't need to make thousands of compounds. We could have, but there was no point because we'd never been able to screen all the compounds. So what that made you do, though, was think really hard about what are you trying to achieve here. And that really came back to what Eleanor said too. Having a target candidate profile was really critical, and that's actually where the experienced people in Exonate were fantastic about working out exactly what do you want this molecule to do. So once we established that it definitely needed to be soluble, but it didn't have to be necessarily stable, or long-term stability meant that we actually were able to optimize those properties differently. And the efficient screening cascade was actually also really important. So we knew that it had to be kinase potent, but then the second thing was eye permeability, and that was a low throughput screen for us, very hard to do until we finally worked out how to do it 10 compounds at a time, And once we did that, we were able to rapidly then work out from our library that we'd already discovered really potent compounds that were permeable. We also discovered lots of not potent compounds that were also permeable, and that allowed us to feed back into how to optimise those properties. Once we had that, we were then able to then tweak the ADME properties to get the ideal candidate. One of the things I did find, though, is that not all medicinal chemistry consultants are right, and particularly if you're an academic, they tend to be a bit dismissive of what you're doing. The first one told me that we should, the compound was too big, too lipophilic, it's never going to work, but I said, but it does go in the eye, right? And he goes, yes, but it's too lipophilic. And I said, well, what do we need to know about this? Well, there's no literature on getting things into the eye, and so he wanted to go off on a different pathway. We sort of stuck to our guns. He disappeared. The new consultant came in who was much more, I think, open-minded about it, but we had a lot of arguments with people, particularly at Wellcome, about Lipinski's rules, and we had to keep trying to explain to them that this is not going into an oral available drug, it's going into the eye. So it's good to keep an open mind, but also stay strong. Last couple of things I'd like to mention is intellectual property. Obviously, filing before you get involved in a company is really important because it does give you some strength if you're negotiating an agreement. And if you're going to use research candidates, you need to think about how do they understand what they're doing in that role and why, and what are they actually contributing. That was always a challenge. The other one is getting research agreements sorted out very early, because one of the things I certainly learnt was clarifying what you have already, what you already know, is really important, because often people assume that they developed it after the agreement started. And certainly what we've found with Exonate, that probably if we could go back and look at it, the negotiating with big companies, they want very clean IP, and so we've got multi-university patents here. They certainly find that very challenging, so it's really important to think about that. And finally, I know people have said they used honour students and PhD candidates. I made a decision very early on not to use them once we decided the company was moving forward, because you can't publish, can complicate the IP issues as well. People get nervous. Much better to negotiate using research assistants and or postdocs. And that's also because timeframes are so much shorter. We know you want to be able to achieve things quickly, and so we found that this was a much more efficient way. Slightly more expensive, but more importantly. And the last thing I just mentioned, that from that experience is that we've then co-founded a new company called Amenda just last year. It's still doing alternative splicing, but now we're going through into cancer and pain as we always originally did. So therefore now we're looking at systemically available compounds. And what I forgot to mention is that Sphinx 31 is actually a compound, has been used against AML, and we've published a Nature Communications paper on that recently, showing that this is a viable pathway if we can find a molecule that's systemically available. Thanks.
Okay, our final speaker for this session is Dr. Paul Stupple, and Paul will be speaking on medicinal chemistry.
So I'm not a member of the Sphere network, so I'm very happy to be here and to contribute and to learn from your experiences today. So today I'm going to tell you about medicinal chemistry at Cancer Therapeutics CRC, and I think I'm going to echo some of the messages that you've heard in the previous two presentations, so it's good that we're all aligned. So firstly, I'll tell you a bit about CTX. We are an oncology-focused biotech in Australia. We identify small molecule, oncology-focused molecules for a portfolio of projects. We're partnered with leading Australian research and biotech institutes. I'm employed by Monash University, but I work exclusively on the CTX, Cancer Therapeutics CRC projects. So I'm not a typical academic. We're very lucky to work with some amazing institutes. We have Peter Mack, who do our in vivo work, the Walter and Eliza Hall Institute, Griffith University, and the Children's Cancer Institute, who do the screening for us. So we have very enjoyable and productive collaborations with Greg Arndt at CCI. So we have all the bits that we need to do to take a drug discovery project from validation of the target all the way through to identification of a development candidate. And the final circle there, we aim to translate Australia's medical research into the next generation of gentler and more effective therapies. So we've got a good track record of success. We are very fortunate to currently be well funded, which means that we can apply a lot of resources to the projects. And from that investment, we've had some big successes. In 2016, we licensed an epigenetic project to Merck Sharp and Dohme for 500 million. These are silly numbers. Upfront payment of 20 million, something like that. And then last year, we licensed 2 chromatin modifier projects to Pfizer for a similar amount of money. And we've licensed other projects as well, not for the same upfronts. So Amplia Therapeutics are progressing a FAC inhibitor that we discovered. Someone spoke about FAC earlier on. They're progressing one of our compounds to the clinic. That's an ASX-listed company. What we've typically done is taken projects to the point of late lead optimization, identifying a set of compounds which are potential clinical candidates, and then licensing those on. I think in future we'd like to take things a bit further ourselves and actually take compounds into the clinic. And we don't have anything in the clinic at the moment, but I think we're optimistic that in the next year or two there are going to be a number of exciting compounds actually getting into patients, and that's what we're all motivated by. So we typically start our projects with a high throughput screen. Our projects are normally novel and in novel areas, and there's not a lot known about them, so we have to do a lot of work to set up the assays that are required to progress those projects. Taking a hit compound which has got some activity through lead generation and lead optimization, and that takes a long time. We've just seen similar numbers, so I've written here from 2 to 4 years through those two phases. And as we've heard, multi-parametric optimization is essential to that process. And you're trying to identify a compound which has got the right activity, safety, PK, stability, absorption, solubility profile. And the compound that satisfies all of those criteria, it's not necessarily the most potent or the most stable, it's the one that's got the best balance of all those properties. So one example of that is the PRMC5 project. This was licensed to Merck and was a project that came to us from Professor Steve Jane at Monash University. So Steve brought us the idea and some validation for the target in hemoglobinopathies. Actually, there was just an emerging story around oncology. But we set up an assay and ran a high throughput screen of 350,000 compounds, took the hits through to dose response, and importantly a counter screen, reordered and retested and saw 77 hit compounds with IC50s less than 10 micromolar from that screen. The next bit that we did was absolutely essential, which was to further characterize those hits through SPR, surface plasmon resonance, which is a measure of the affinity of the compound for the target protein, and also cellular testing. And from those 77 compounds, we found just one that worked. At the time, we didn't have a high throughput cellular assay to progress all those compounds through. We just took the best ones through low throughput in vitro cellular assays using Westerns, and that's how we found our top compound. We later went back and checked that we hadn't missed anything. But yeah, a very small number of compounds, just one chemotype, was identified from that screen, and this is consistent across a number of our projects that we don't have high hit rates of validated hits. So this was the hit compound, 262135. Really fantastic cellular activity for a high throughput screening hit, 45 nanomolar in a cellular biomarker assay. And structurally attractive as well, not too many features in there which caused us many concerns. And that hit was found in November 2012. We went through the optimization process and we made, or perhaps tested, I think is a better a word to use, 1,000 compounds to identify 2 chemotypes, and one of which is represented by 393293 or L1352 in the Merck nomenclature. And it had subnanomolar activity in the cellular biomarker assay. In the interest of time, I'm just talking about potency here, but obviously I think the major challenges for this chemistry lied elsewhere in terms of optimizing the pharmacokinetic profile, which can be very expensive. We're very lucky in that we had great support from CSIRO with the crystallography, and so this was a structure-based project which was essential to accelerate the progress of the project. So the project was licensed to Merck in January 2016. And just to echo another point from Jeff, actually, a company in the States, Epizyme, and GSK actually, identified very similar hits to ours and published those in a series of patents, but also in an accompanying Nature Chemical Biology paper. And so we were second, and it really ignited interest from big pharma in this area. We had 5 companies, five big pharma companies, all wanting to negotiate on this project. So that resulted in very favourable terms for us. But I think it highlights something important, and that's that often being first isn't absolutely essential. It's often really good, but if you're second, then that can be good too, because big pharma often follow their competitors, and this worked out very nicely for us. So we are not large enough to obviously do all of this on our own, and we've used a lot of CROs in the process of identifying the candidate. I'd like to do a little advert for the Centre for Drug Candidate Optimisation at Monash in Melbourne, which is run by my colleague Professor Suchting. They're fantastic, and they do most of our in vitro and in vivo ADME. So I've listed the assays that they contributed to this project there, which are numerous. But in addition to those, we've used a number of companies for in vitro safety testing, and cell line screening and dog PK at Wuxi. So these are an essential part of the drug discovery process. I wanted to make the point that we don't always find 40, 50 nanomolar cellularly active compounds following a high throughput screen. So I've put 2 other projects which we have progressed to compounds that we feel have the potential to be development candidates. Both of these were run both these hits were identified by high throughput screens. Project A was a phenotypic screen where we actually had a target in mind. So the first assay was an SPR assay for that particular protein, an approach which I really like because it gives you the opportunity to find new projects from your high throughput screen. And the hit was 190 micromolar, and we optimized that down to 120 nanomolar. So the point there is that some of your it may be the case that your hits can be very weak. Project B, more typical activity or affinity from the high throughput screening hit, but we optimised that to a 20 picomolar compound. So the point there is that huge gains are achievable in this process. So how do we go about this? Our approach has kind of evolved over the years, and there's now very clearly two steps to it, and that's design and synthesis. And I've put my team in Melbourne there, Ilva, Ben, Michelle, and Jonathon. Fantastic group of chemists, and they are very much responsible for design and project leadership of the CTX projects, and synthetic problem solving as well. The bulk of our synthesis is now done in a CRO. We currently use Synthesis Medchem, and typically we would have 5 to 10 FTEs for a lead optimization project, together with perhaps one FTE or perhaps 2 FTEs from the Melbourne design team. So those compounds are made in China. They come back to us in Melbourne, and they go through our screening cascade. And there's a picture of Brendan Monaghan, who's our fantastic CSO. So my last slide is failure. I think this is the first time I've ever had my final slide with such a negative title. But I think it's important for all of us to think carefully about it, as other people have mentioned. Failure of drug discovery projects is to be expected. If you're going to fail, then fail quickly, as one of the former presenters shared, and prioritise and invest in the go, no-go experiments. In terms of the risk of failure of the chemistry, you can reduce this through ensuring you've got the best hits, that you validate your hits in terms of identity, and also putting them through orthogonal assays. I can't emphasise that enough. Avoiding structural alerts, investing early in understanding DMPK and safety profiles, and trying to identify contingencies.
I'd like just to direct a question to Greg. I'll just start things off. With respect to miniaturisation of high throughput screens, I mean, I'm reading that we can go down to 1 microlitre quantities, possibly even less. And I just would like to know, how do we manage research quality over this vast quantity of screening?
Yeah, that's actually a very good question, and it's something that we have taken a lot of time to build into our HTS facility itself to actually deal with the fact that we're now moving to more miniaturized assay formats. 384 well formats are generally what we do, but we're now moving to 1536 well formats. And of course, you need to have back-end database management, which we have. You have to have informatics to be able to track all of those compounds. It's not a simple thing to do, and so having an HTS facility which has all that built into it is absolutely essential if you're going to do that kind of miniaturization.
I think it's excellent. Congratulations on setting that up. And also, I guess, managing system failures as well. I mean, these machines are just running 24/7.
Oh, absolutely. So running an HTS facility with all the automation workstations, we have 4 of those at the at UNSW right now, managing those workflows, but also looking at ways in which you can integrate all of the different workstations to optimize the number of compounds that you can actually screen.
Excellent, thank you. I'd like to open the floor now for questions. Yes. Can we have the microphone down here, Tanya, please? Thank you.
Hi, for whoever wants to answer these questions. So I am currently at the moment on the optimization part of the screening. And I just wanted to get your input on how do you know when to stop, basically? Because it looks like it can just, like, I guess at some point there needs to be a decision, and how do you make that decision?
I could just field that very quickly. One of the things that we found working with a lot of researchers in terms of assay optimization is it's probably very important at some point that you contact and get advice from people that run these kinds of facilities. So there's ours, but there's about 8 or 9 across the country that actually do this. And we can sit down with you and go through the optimization process that you've done and give advice as exactly that. When do you stop? So there's a certain number of experiments you should do once you have your assay at a certain position in the optimization process, and we certainly can advise on that.
I have a question for the medicinal chemist. So Tristan, if you can please introduce yourself. Tristan's over here.
I'm Tristan from UTS, medicinal chemist.
Right. So for the medicinal chemists, we're in an evolving R&D landscape, right? So biotech drugs are really coming to prominence in pharma. So where can medicinal chemistry, which has been the bread and butter of pharmaceutical industry, how can that add to this evolving new world, I guess?
Sorry, evolving in terms of?
In terms of the biologicals taking dominance, coming to dominance. Yeah.
Perhaps someone else can answer that. I'm the small molecule guy myself. Yeah, I don't work in the biological space. I see it as a great potential, particularly in cancer. As a small molecule person, it makes me a bit nervous. But yeah, I don't know if anyone else has experience in biologics.
Yes, thank you.
I don't quite know where to start, but I think things are changing all the time. You know, for all the disciplines involved in drug discovery, and I think at the moment there are some big questions around new modalities. You know, for example, PROTACs. And for those who are unaware of what a PROTAC is, it's where you connect a ligand for a particular protein with something which is recognised by the ubiquitin ligase system, and you can effectively reduce the levels of a protein in a cell, or completely remove the protein from the cell. So there's massive investment being made in this at the moment, and so who knows quite where that's going to end up. I mean, on paper, when you see the publications, it looks like there's a formula that you can follow to identify, you know, your potential drug if you've got a known binder for that particular protein, and then the phenotype that you would see from that probably is more likely to mimic what you would see from a CRISPR experiment. So I think that's an example of you know, one particular new direction for medicinal chemistry.
Fantastic.
Yeah, and I don't know what else is around the corner, but no doubt there'll be something.
Great, thank you. That was my next question, actually. What's the next step for medicinal chemistry? Yes. Sorry, Phoebe. Yeah.
A couple of questions. One is, are all CROs the same, and how do you select? And the other question is, your number one candidate that come through as your hit is highly insoluble, and maybe Martina, you want to touch on the potential of nanomedicine as a delivery agent.
Okay, yeah, so I basically make nanoparticles to enhance the delivery of these drugs, and Lipinski's rule goes out of the window. So we can basically deliver drugs that are highly insoluble. We can deliver drugs that are very big. But what we really need to do, we need to work with the medicinal chemists, with the cancer researchers to really understand the problem. So nanoparticles can they have usually plasma half-lives of an average 10 hours to 1 day. So they circulate very long time. And I think there's a good place for these kind of drug delivery vehicles. Of course, they have issues by themselves. What do you do with an empty drug delivery carrier? But I think the opportunities are certainly there to really look at solutions of drugs that get otherwise discarded because they're so insoluble.
And CRO selection? On anyone on the panel? Yeah.
So I guess I've been fairly lucky in that the first CRO I started working with when I was in industry have been really great, and we've stuck with them now that I'm back at UNSW and working for TroBio. But I think also it's, for me as a chemist, I think having a chemistry team that are really communicative. They have, and it's not just our regular teleconference every week. It's also, emailing me if there are issues, and
it's really felt more like a collaboration. So even though I've been doing all the design and just giving them lists of molecules to make, in terms of like problem solving with the synthesis or troubleshooting, trying to identify an impurity in an NMR or something like that, it's been a real sort of back and forth and a good collaboration. And I think that's something to consider as well. Just here, a good working relationship, as well as just considering how much does it cost, how fast is it.
So I've got a question about the libraries, publicly available libraries versus libraries that aren't publicly available. How important is that in the process? Because it seems to me if you get a compound that's publicly available, you've got to put quite a bit of work into making one that isn't.
So there's a lot of back-end work that goes into actually designing a novel diversity chemical library. And so, for example, the ELID library, the 300,000 compound library that I was talking about earlier, really was built on not only drug-like and lead-like molecule structures, but also getting rid of what are called pains. Those are those molecules that always seem to come up in your screens and are actually just promiscuous and or just a pain. That kind of design has to go into the library, I think, which then provides you with a higher probability of getting something novel that you can work with, good chemical matter to start with and start the medchem process. That's my view.
I think it's important to remember that a hit is just a hit. I mean, it doesn't matter whether no one else knows about it or not. Ultimately, it's going to require medchem optimization. You're not going to get a drug from your library, particularly if that library's been sold to lots of different people anyway. So I think that's the bit that a lot of biologists struggle with, is that there is going to have to be this iteration of optimization. And that comes back to understanding what do you want the molecule to actually do, like how soluble, etc. So just view the hit as a starting point.
Jonathon, I'll continue on the questioning, if I may. Your presentation, absolutely superb. I mean, that was a fantastic story, as we say. I was very interested because around the patenting and being contacted by, and I happen to know Dave Bates, so I've been at the University of Nottingham, so I know, and I'm not surprised that he actually did contact you. And again, I think probably there may be a couple of us in the room, maybe a lot in the room, who've been involved in projects over the years or seen projects over the years where the opposite has happened. And so people have filed a patent with their home university, and I think we all know that just leads to massive arguments that a lot of them never get resolved. And so I think I just wanted to hear, I guess, the positives of the story with that contact, and just sort of seeing how you've made what really is and a series of startups, because we've now seen this later one that you've started as well. And I guess, were you surprised, and how did you sort of deal with those early discussions when Dave contacted you?
Yeah, it was certainly an interesting process. I mean, David Bates is a fantastic person, so I mean, he was always interested in working together and doing it. I guess the patent, I wasn't even thinking about patents at the time, and certainly with Stefan, he works in open source drug discovery, so I hadn't even, I guess, contemplated it. So it just came out that Dave had this special circumstance where he was able to have some funding to file a patent in the first place that he thought he could actually use. So the only thing that was odd for me was the founding of the company, because that sort of happened rapidly, and with hindsight, I wasn't engaged in that conversation early for Exonate, and that was just one of those things that happens. Whereas the second company is with David again, and we've done that, I would say, properly this time, that we've all sat down and worked out exactly what are we trying to get out of this. But I mean, the beauty of that one was that I was the medicinal chemist and he was the biologist. So, and I guess the big thing was all of my molecules blew away every one of his molecules that he already had. So, and he was smart enough to know that if he tried to file something without my involvement, then obviously it'd be invalidated. So, but yeah, I think what I learned from that with everybody I talk to now is, let's be very clear about what we're trying to achieve, and have those patent discussions early about and research agreements, and then at least everyone's on the same page.
Dave.
I wanted to ask Eleanor and anybody else on the panel, I mean, one of the big problems at phase 1A is actually the unexpected off-target. So I like your fail-fast model, but what are you thinking about fail-fast preclinical to reduce the risk of off-target?
Yeah, that's something we'd definitely consider, because you want to find out something's wrong with your molecule at the preclinical stage. So I think you can. I think a really good design of your in vivo tox, really sort of interrogating what's going on when you hit those higher doses, I think is really useful. In terms of, I mean, there are an awful lot of proteins in the body, and you could be, potentially hitting a whole suite of them. So I think there's always going to be that possibility that there's something you haven't thought of that's going to come up in the clinic. But I think at least I think going for like the obvious anti-targets, things like HERG, and also your related proteins is really important. But I guess a lot of it, you're going to have to look at, say, in vivo models to say, what do you hit? Try and figure out what you might be hitting that you weren't expecting to hit. And you're not going to see that just trying to test for everything individually. I think putting it into an animal and seeing what happens is probably going to be your best sort of risk mitigation before putting it into humans.
Can the panel comment on CRISPR knockouts and whether you think that that's relevant? Can the panel comment on CRISPR knockouts? That was one of the things that in our instance, we did a CRISPR knockout, which obviously it's a problem if the cells need it and you can't actually make a CRISPR knockout. But if you can, that's a good way of testing, and that was one of the issues that we had, where we could still see anti-cancer effects in the knockout. So, obviously 5 years ago this wasn't something you could do. Now it is. Is that an issue moving forward? Because just because you might hit something else, it's not necessarily a problem, but it causes a great degree of nervousness moving forward if you've got additional effects.
Yeah, so the inherent functional redundancy that exists with some targets, how do you address that? And I guess that's what you're asking with the CRISPR knockout.
I'll go back to where you started, which is talking about CRISPRs and how relevant the data is. I think it's really good data to have at the start of the project, and a real step on from what we were all doing before that. However, we've seen it time and time again where the phenotype that you see from CRISPR is completely different to what you see from pharmacological inhibition. So I'm a simple chemist. I don't understand all of these things, but I say to the biologist, can't you just mutate the binding site so that you've got a catalytically inactive protein? For me that's fantastic data if you can get that. But I know that's a lot harder. So that was the first part of your question, and after that I'm not.
Was also using that as a kind of counter screen. So you've got a CRISPR knockout. Does your drug still do something?
Yep, that sounds good. Yeah. We don't do that that much. Yeah, sounds like a good approach to me.
Yes.
Yes.
Ask the question.
Sorry.
Yes. So we have a target that relevant to pancreatic cancer. So now we're trying to find the first compound. So I heard that this new technique called fragment-based thing, and also the old way. I mean, what is your recommendation?
So I can only really talk about sort of in silico. If you have ideally a crystal structure, or at least a really good homology model of your target, you can then I've done sort of fragment-based drug design from docking. So basically you dock really small sort of chunks of molecule and then sort of see where they fit, and ideally you'll have one chunk that fits in one part of your binding site and then another chunk that fits in another part of your binding site, and then you can figure out ways to sort of chemically connect them. Or you can take something very small that seems to bind and then sort of expand on it.
I know that you can also look at fragment-based, like actual screening with the protein, but I don't have as much experience in that. But the idea is you find something very small that might bind very weakly, but because it's small, you've got room to grow. So you would then get a medicinal chemist to, usually with a structure-based approach, like build out into a bigger molecule.
And can I just add to that, as Eleanor said, there fragment libraries actually exist. I know MIPS has a library that everybody's using, and we can do those kinds of screens as well, physical screens, as fragment-based screens rather than in silico.
Yeah, we're doing it with
Oh yeah. So you're doing the screen here through Sydney Uni. Yeah, and I think they actually use the Monash library as well.
Yes, the same from the Monash library, yes.
I think one last question over there.
Thank you. So Anita Vandermeer from Clinical Trials New South Wales. I just wanted to pick up a point that Eleanor made in her talk about beginning with the end in mind, and perhaps ask Jonathon and Paul if they would describe a little bit about their experience in the licensing with Big Pharma. Moving beyond just the science itself, what are the issues that need to be considered when you're starting to look at licensing engagement with Big Pharma?
Yeah, well, obviously it's really important. What we found was the IP has to be really clean, right? I mean, that was the big challenge, was all these different university agreements with the molecules in the first place. So that was, one thing that certainly we had to be careful about. But in terms of, are you talking about the actual molecules themselves, or? More about the commercialisation pathway for engagement with Big Pharma. Yeah. I know the IP is one issue. For example, is the length of your IP, how long you've been sitting on an issue for pharma? Yeah, okay. Something they want? Yeah, certainly, I personally wasn't involved in that sort of stuff, but you find that you're having these conversations about what is, what IP do you hold? Does the company have a license for it? Can they actually, get a long-term benefit from it? I think that's a really important part. I might hear you've done.
So, in terms of what from our experience, what Big Pharma are generally looking for, the point at which they get excited is when you have an in vivo active compound. So we have ongoing conversations with Big Pharma about our projects, and they just want to hear updates all the time until you get to the point where you've got a novel target, in vivo efficacy, and then that's when they get more excited. There was a point earlier on around understanding the target of your small molecule. We've had one of those. We've had a project where we didn't know the target. We had a lovely phenotype, no interest in that. So that feels that's been quite important. That was quite important for that particular example. When you have the interest of Big Pharma, the process is slow. It takes a year from when you think you're going to do a deal to actually doing it. And there's lots of backwards and forwards and agreeing terms, and it's a stressful experience because if you're sitting on IP, then you're nervous the whole time that patents are going to publish, which blow you out the water. So that's not good. I think that's everything from me.
I think we have time for one more question. So if anyone has a question. Back, please. Yeah.
Thank you very much. The CTX model has been incredibly successful, and also in Queensland you hear of some fantastic success stories like Spinifex, and I'm just wondering, we're not quite at that stage in New South Wales. What is it that we need to do to get there? Is it purely just investment, or are we missing something here?
Well, I can tell you that I have never had my XNATE work funded before I started it. I applied for NHMRC grants. I mean, medchem is expensive and hard to do, and you're in the sort of valley of death between the ARC and the NHMRC. So I think that's something that is hard, and also getting biologists to understand that you do have to give a chemist some money to actually make some molecules is... So if you go back and look at Queensland with Spinifex, that was there was good investment there from the university to actually allow the medicinal chemists to start that work. And CTX is the same sort of principle. It's got that core, I think, of being able to get things going. So in my case, it was I fortunately had moved to UNSW fairly recently and had some startup funds, so I was able to invest it in a way that I thought was sensible for me. But yeah, when you get a new project, it becomes that challenge of trying to do it fast enough. If you don't have enough money, then it becomes a challenge.
So, do we think that it's state government as a community that we need to approach?
I think there's certainly those discussions are happening now, but yeah, I think people realising they do need to put some investment into that level, because I think we're quite good at target identification and validation, and quite good at taking other molecules that are finally there, but actually the number of people who do high throughput screens who just think they're going to have a drug in a week or two, I mean, is very challenging, right? I mean, so it'll take time to do that, and I mean, as Paul's saying, he's got 5 to 10 FTEs working on in CRO. I mean, we only ever had 1 or 2 smaller company, but it's still a significant investment that you have to do to actually get enough compounds to find the right one.
And I just comment, it's a little bit to do with what we're developing in our therapeutic innovations group, is to try and find ways in which we can actually focus our attention in that area that you're talking about, because we've had so many projects where people have got hits and then they just disappear. And what we should be doing is putting some effort into that actual area and driving the medicinal chemistry for lead gen and lead op. That's my view.
Greg, you mentioned UNSW. Is it available across the Sphere network, your high throughput?
Yes, available across the network. We're willing to talk to anybody. We're at the stage now of really developing the process, and I'm sure we'd like to have to and fro with people that might be wanting to partner with us and collaborate.
Okay, thank you to our panellists and to our speakers. Please join me in thanking them. So our first speaker is Associate Professor Mark Cowley, and Mark will be speaking to us about companion biomarkers, including omics.
Thanks for the intro, Mary. Very big thank you for the invitation. I wasn't sure how to phrase this today or how to pitch this today, but it was an invitation given after I do my usual spiel of sequencing lots of kids with cancer, finding mutations, and some of that has some pretty important implications for those kids. So I'm really going to try and do a similar pitch today. We've got 10 minutes to really tell you about some of the work that we've been doing that helps identify druggable targets in patients with cancer, and how we can try and interface that with drug development. I really enjoyed the earlier sessions. It was really eye-opening about what you do after we identify these targets. So I've had a really good time so far. So really the context that I think about most of cancer at the moment, at least, is in the context of precision cancer medicine, where we are essentially doing a whole suite of molecular profiling techniques to understand the molecular drivers of patients with cancer. Originally that was pancreatic cancer, then moving on to adults with rare tumours, and now onto paediatric cancers, which by definition are rare tumours, but we're focusing on the ones that have really high risk, really poor outcomes. And so what that really means is it's challenging, because every day we've got different tumour types coming through the door, and you can't be an expert in all these different disease types. So genomics is a pretty good lens to try and interpret all that information with. So essentially, the point of the slide is that we're trying to use precision medicine studies not only to help the patient, but also to generate an awful lot of information for rich research value and identifying new drug targets, which I'll talk about later. A little bit of context about paediatric cancer specifically, and because we're talking about target identification, is that kids are not little adults. Some of them may have tumours that sound the same, but they're inherently different tumour types. This popular graph just shows that the tumour mutational burden is often lower in kids, 10 to 100 times lower. There are fewer mutations which drive these tumours, which is actually an opportunity to really tease apart what that one driver is doing on a patient. Importantly, though, only 45% of pediatric cancer driver genes are found in adults. So we've got different targets that we need to be looking for, which means different molecules to be designing drugs for. And importantly, a lot of the driver mutations are copy number and structural variants, not SNPs and indels, which seems to be a little more common in adults, and also helps justify our choice of quite comprehensive molecular profiling, which I'll talk about more in a second. Even when the same genes are mutated, often different mutational hotspots are mutated. So as many of us are starting to think about liquid biopsy and screening the blood for these mutations arising in blood, we may need to be looking in different parts for pediatric cancer as opposed to adult. So different targets. So I'm sure many of you, especially in the Sphere group, know that the Children's Cancer Institute, in partnership with Sydney Children's Hospital Network, are leading the Zero Childhood Cancer Program. It's a fairly ambitious national program to recruit all kids around the country with high-risk paediatric cancer. And for that, we define high risk as patients with less than 30% chance of survival after 5 years. We're in the second phase of the Zero program called the PRISM National Clinical Trial, and we started off with a target phase called, Target, which was our pilot program where we developed the molecular profiling techniques. The major take-home message from this slide is that we're applying a really comprehensive approach. We do deep whole genome sequencing on every patient, RNA sequencing, methylation profiling on the brain tumour and sarcoma patients, and increasingly moving into liquid biopsy as well to track these patients longitudinally. I'm a very, very big proponent of as comprehensive a profiling as possible, especially when you're being thrown rare disease patients, leukemias who've failed all standard therapy. The tumours teach you something different each time. You usually find something that's been missed earlier on in the treatment journey. And so we're really trying to make the case that these profiling techniques can be really important. I'm not going to touch on Greg and Tim's work in drug screening, which is a companion part of this program, and a very important part of this program as well. Right. In a slide, I normally have lots of anecdotes about finding mutations that work. Here's my favourite. This is a 11-month-old girl who had a tumour the size of her chest tumour. You can see in the top left there in a little orange outline. In 9 days, we knew she was extremely sick in palliative care in hospital. And in 9 days, we managed to sequence her tumour with our partners at Garvan Institute, run the analysis on the cloud through pipelines that we've developed, and find a novel NTRK fusion. This NTRK fusion had never been seen before with a SPEC1L partner, and so the pathology lab missed it. No criticism there. They look for the common NTRK partners. So importantly, this helped make the diagnosis. And a couple of weeks before we sequenced this case, this was 2 years ago now, ASCO had occurred. Many of you will know the remarkable success story of Loxo Oncology, who've developed larotrectinib. It's an amazing new wonder drug. We were able to get the very first compassionate access to that drug in Australia. 2 weeks later, the patient started responding, and 2 years ago, we just resected the final little part of her tumour out a couple of months ago. So she's disease-free. Great, cool story, but it doesn't happen all that often that you get a single agent curing a patient. Maybe it's even too premature to say cure, but very long-lasting remission, can we say that? Larotrectinib, amazing new wonder drug. So the colour of this waterfall plot is the type of tumour that the patient has, so the drug doesn't really care what the tumour type is. Many patients seem to respond to it if they have the NTRK fusion. Doesn't seem to matter which type of NTRK fusion. And this was, I guess, a bold, I don't know how bold, but a decision by Loxo to develop a kinase inhibitor very, very specific to the NTRK clade of the kinase tree that you see there on the left. Importantly, this is the first FDA-approved targeted cancer agent based on mutation rather than cancer type. So this was a huge achievement, right? This is now a companion biomarker that detects NTRK fusions in patients, could theoretically make this recommendation in any cancer type. Now, if we had to wait for the companion biomarker to have been developed, the patients wouldn't have accepted them or benefited from them. So that's, again, why we're in favour of a comprehensive screen. But I understand that as this gets closer and closer to being rolled out globally, a companion biomarker that looks specifically at NTRK fusions would be a great test to find patients that would respond to this drug. Sorry about the colours. I did this very late at night and pasted it across, and the colours are a bit gnarly. But essentially, the essence of this slide is that when you look at patient whole genome sequencing data of the germline, the tumour, the RNA-seq of the tumour, the methylome, there's dozens of different analyses that you need to do in order to find all the potentially actionable mutations in a patient. And in any one patient, it could be a germline mutation, or it could be a somatic, or it could be a hypermethylated gene. And so you've got to be on your toes to be able to find all those different markers. And I think through Zero, we've been able to build some infrastructure to identify a lot of them. There's still some points in here that we would like to do better, but that's been able to identify a really large rate of actionability in these patients, or sorry, I should say, identify a large proportion of patients with a potentially actionable drug. So I want to thank Loretta Lau, who's a precision oncology fellow, who helped me put some of these slides together summarising the clinical side of what we do. So if we take 179 patients that we've discussed at a molecular tumour board, we've made a recommendation in 68% of those. We just had our annual review meeting about PRISM, and we thought that would be 20, the royal we, thought that might be 20 to 30 patients that would benefit from this type of program. Instead, 68% are actually getting a benefit from this type of profiling. So we've made 212 recommendations over these 122 patients. Some of those recommendations are now for combination therapy. And that's why I asked the question of the panel earlier, is that I think we're moving into an era where we can't just make single agent treatment recommendations and hope that it's going to work all the time. Importantly, and something new for our program, is that a quarter of those recommendations are based on outlying gene expression. So really overexpressed genes in the absence of supporting copy number change. That's been quite a journey to convince the oncologists that some of that information is important. The idea being that if it's overexpressed, it's temporal and it could drop back down again. But we've had some really good treatment responses to that. We've had to develop a 5-tier treatment recommendation way of recommending these things back to the clinicians, because some treatment recommendations are stronger than others. So that's shown there on the left of the slide. Tier 1's our highest tier of evidence, where we find clinical evidence of that recommendation in the same cancer type. Tier 2 is other cancer types. Tier 3 and 4 are preclinical evidence. And Tier 5, really hypothesis-driven research stuff that has to pass the muster of the molecular tumour board and all the clinicians in the room. So evidence-based. You can see from the middle pie chart that most of the time it's a single recommendation, but for some patients we're making multiple treatment recommendations. The take-home message, this type of profiling can identify things in more patients than we expected, and it's been quite that part's been quite successful. I don't want to get too stuck into the details, but I thought if we're talking about targets, some people in the audience will love a slide like this about the types of genes and pathways that we're finding mutated in patients. So PI3K, AKT, mTOR is the most frequent pathway that we find affected. You can see in the third column there that there's a whole range of different molecular mutations underlying those particular recommendations. Sometimes it's a mutation, sometimes it's a copy number change, a gene expression change, a fusion, high tumour mutation burden. All of these things have high clinical utility in terms of making these recommendations. Some of these are more druggable than others, and you can see that some of the ones with inhibitors are shown in the pie chart there. And obviously the receptor tyrosine kinase category there is quite a rich category where we can find lots of treatment recommendations for many children, not always with enough evidence to be a blockbuster recommendation, but often enough to make it back to that patient's molecular tumour board report. Interestingly, what's in my mind a lot is combos, stop doing single agent treatment recommendations. So there are a number of patients with alterations in multiple pathways, and this might help influence how we might design the next phase of PRISM 2, or whatever we're going to call it, to help have novel clinical trial designs to help target multiple driver mutations, let alone when you start combining what Lisa showed us in the first session about, was it statins as well, and different agents that really understand the biology of the cells that you're trying to treat. So really, I've tried to stick to time for once in my life. So the take-home messages from this talk really are that kids' cancers are different to adult cancers. They're driven by different genes in many cases, different mutations, and we often don't have a pediatric dose for many of the drugs.
we make more treatment recommendations actually than what I've shown, but if the oncologist can't find any pediatric dose for those recommendations, they don't go back to the patient, which is disappointing and leads you to think about PK studies to try and work out how we can bring those drugs, those good drugs developed in adults, into kids. I've only showed you one anecdote, but there are dozens about comprehensive molecular profiling, identifying lots of companion biomarkers to help make these recommendations. As I said earlier, if we take one tumour and we have to run 15 different companion diagnostic tests to work out which one the patient might have, you've spent a lot of time, money, used up all of your tissue, and if you can do one comprehensive profiling approach, then we believe that that's going to be the way to go. We're trying to gather the health economic data to convince government that that's also the way to go, particularly when there's only about 2,000 kids being treated for their cancer at one time. It's not an enormously large number that you can't think a fairly expensive test like a whole genome is not appropriate. Larotrectinib is awesome. We've got 8 patients on larotrectinib right now, and it seems to be extremely effective. Doesn't matter what the cancer type is. And how do we do combos? How do we develop a clinical trial framework such that your patient can tell you what combinations they might need to be treated with and deliver that in a meaningful time frame without being drowned in all the paperwork of opening lots of subarms on a treatment study. I'm very remiss to not have an acknowledgement slide. I did this very early this morning. So this is big team science, lots of funders, and I really want to thank the team for helping what we do. Okay, cheers.
Our next speaker is Dr. Steven Suchting, and Steven will be speaking to us about intellectual property and commercialisation. So please welcome Steven.
Thanks very much. Thank you for the invitation to talk. So I work for a company called IP Group, and for those of you who don't know, we are an investor in early stage technology-based companies, including therapeutic companies. I work for the life sciences team, and I was asked today to talk a little bit about commercialization, and I think we've heard a lot about the mechanics of drug discovery. And I'd like to talk a little bit about the commercial aspects of it and why they are so important and why you need to be thinking about those really from the initiation of any sort of drug discovery project. So first, a little bit about IP Group. So we're a UK company that was founded about 20 years ago. We are, unlike other venture capital firms, we're unique in that we're actually a public company and we're listed on the London Stock Exchange. And over the past 20 years, our model has really been to partner with universities and to invest in very early stage technologies that are arising out of university research. It's been a successful model, and we've recently expanded into a number of other countries, including Australia. And currently, our portfolio is about 150 companies, with about £1.2 billion of value that we own in those companies. And actually, one of those I learnt just today was Exonate, which Jonathon spoke about earlier in the morning session. So, what do we do? So, we really see ourselves as a partner to really bridge the chasm between innovation and commercial success. So you guys are over here doing the innovative research, but often struggling to bridge over this valley of death, as it's known, to really achieve the commercial success. And this is what we come in and what we think we can uniquely add to help achieve that. And we do so because we have, I think, very deep scientific insight. We're very much based around the science and the underlying technology. We have access to permanent capital, and I think this is a distinguishing feature. We're not driven by having to get an exit on a certain time frame like many venture capital firms are. So we can actually invest in early stage companies and stay with those companies until they're at the right maturity to exit. And we have a lot of experience building companies and also providing the support infrastructure that helps achieve success over the long term. So in Australia, we've recently entered into the market here, and we've made a $200 million minimum commitment to invest in new companies over the next 10 years, predominantly coming out of our partners with the group of 8 universities, including UNSW and the University of Auckland. And we have formal partnership agreements with those group of 8 universities, which enable us to have very early stage access into the disclosures and IP coming out of those universities. We're covered by confidentiality, and we have pre-agreed terms for spinning out companies that really streamline the process. We are not exclusive to those companies, to those universities, and we will also work with pretty much any academic-based institution. So if you're outside of UNSW, don't feel like you can't talk to us or you can't engage with us. We've engaged heavily with institutional investors who invest both in our portfolio companies and into the PLC in London, and we've spread out across 4 cities in Australia, and we've had a pretty successful year to date. We've got 5 portfolio company investments that have been announced so far this year, and 3 more which are coming in the next couple of weeks, including Inossi Therapeutics out of Monash and Jetra Therapeutics out of University of Queensland. So you've probably all seen slides like this, and what they illustrate is that the drug discovery process is long. The drug discovery process is complicated. It has multiple steps, and it's going to require multiple partners and stakeholders between when you start off and when you actually achieve a drug that would reach the market. Most academic groups and researchers are down this end of the spectrum, and this is predominantly where you're going to be starting off. What you find as you try and progress further on, it becomes more and more challenging. And the reason for that often is that you guys are limited by access to the type of infrastructure that you have available, the type of funding that you have available. And as you get further down this pathway, the drug discovery process becomes more and more expensive. So what you actually need to realize is that to get past these phases, you're going to have to engage not with public funders but with private funders, and they will usually be in the form of pharma companies or venture capital investors who are going to invest into spin-out biotech companies. And in order to then justify putting at risk a lot of private capital, you need to have the commercial case really upfront and well thought out from the initial outset. Because if you look at this slide, what you see is 3 take-homes. Drug discovery is expensive, drug discovery takes a long time, and it has a very high attrition rate. And if you think that you've got to put private capital into something that is expensive, won't get an outcome for 12 years or more, and has a 90% failure rate, really the commercial considerations are paramount. And so they're often, I think, in academic groups pushed to the side because you're so focused on the science about can I do it, you never stop to think about should you do it. And so I thought it might be useful today to have a little talk about what we as an investor would look for in a therapeutic project and what we do to evaluate those projects. And they really come down to 5 pretty simple questions. Is there a market opportunity? Can you get a competitive advantage? Can you generate a financially attractive return? Do you have a backable team? And do you have an executable development plan? It's not rocket science, but it's something that you need to start thinking about really at the very first outset before you get too far down the pathway. So market opportunity really for us comes down to clinical need. What is the indication? How severe is it? What's the prognosis? What's the progression look like? How many patients? Where are those patients located? How easy is the disease to diagnose? What are the biomarkers? How do you stratify those patients? What's the impact of stratification? Because with most targeted-based therapies, if you stratify the patients, you reduce the number of patients that you can actually access. And what's the current gold standard of care? And really that defines what the unmet clinical need is and what the gap is that you need to bridge to get an attractive type of therapeutic. And without really overstressing it, this is really the critical part. You have to, you're trying to address an unmet need. You really have to be honing in critically on what is the addressable patient population for that unmet need. It's not good enough to just say cancer or, even breast cancer, you have to hone in onto a particular subtype in most cases. We also look at market, both in terms of the market size, particularly in the US, and how is that market going to change over the next decade, because it's not going to, any drug you start now is not going to get into the clinic for at least 10 or more years. Can we achieve a competitive advantage? And here I want to stress differentiation, differentiation, differentiation. It's the key factor that I think most academic drug discovery projects fail on. And they fail on it because as academic researchers, you are focused around your science and how your science is different from everyone else's science. And too often you don't really think about, I think clearly enough, that differentiation comes down to the patient. So how is a drug that's going to make it to the market going to be different and differentiate from what's there? How is it going to be more effective with patients, or is it going to be safer? And efficacy and safety are the two primary drivers, but you can have other areas of differentiation around dosing, compliance, and route of administration, but it's all focused around the patient and the clinical aspect of the drug, not necessarily around the science. That said, if you do have a new type of target, the scientific underpinning and the scientific rationale is going to be critical. So we're going to be looking at things like the disease linkage, the target validation, the mechanism of action. These all have to be understood, or at least you have avenues for exploring how they're understood, for us to really see whether those patient-based differentiation factors will make sense. So it always comes back to that idea about how is this going to make a difference to the patients. And also, how is it going to be different from the competition and the pipeline? And so competition is both marketed drugs that are currently being used in patients, and it's also the pipeline. And so the pipeline can be both for the target or indication that you're looking at, but also for the target or the indication that you're looking at. And so we're going to look at the pipeline, what stage in clinical development those competitors are, and if there's a good competitor in the pipeline, how are you going to be differentiated against that competitor as well should it reach the marketplace? Is the financial return attractive? And so for us as an investor, what we're looking for is the value in the program. So we're going to be looking at what is essentially going to be the drug pricing. Are there going to be challenges around reimbursement? Are there going to be challenges around regulatory approvals? What's the potential peak sales potential? All these factors that drive the value. And I think the other area of value is the strength of the IP position. So patents are primarily thought of when we think about IP, and it's the strength of the patents. Is it composition of matter? How broad is the patent and the scope of that patent? How long do you have in terms of the patent life? Because every day at the end of a patent for marketed drugs can be worth millions and millions of dollars. So patent life has a direct impact on value. But it's not just patents, it's the data. And the data that can form the basis for regulatory submissions, materials, methods of manufacture, these all combined come up into an idea of the IP. And then counteracting that is the cost and the time of the development program. How long is it going to take to get to the marketplace, and how much is that going to cost? And so that's sort of the value, and that feeds into the potential exit. So as a biotech company or a spin-out, you're never going to be able to take something to the market yourself. You're going to have to license or partner with a pharma company. And so we look very closely at what sort of deal activity is currently happening for that indication, in that target, in that modality. We're looking at how many deals pharma are doing, what's the size of those deals. And this all feeds into sort of a returns analysis that we'll do, that we get a sort of what's the time cost of capital. And to really put it in perspective, for a venture capital firm like us, we're looking for things that can be, $100 million sort of exits. And to generate those sort of exits, you need to have drugs that are going to be worth, hundreds of millions, if not billions of dollars. And so that's the sort of commercial consideration around financial return you should be thinking of when you start talking about, don't go down a spin-out route. We look very closely at the team. At the early stages of a spin-out company, the academics are going to be the team, and so we want to see, the experience and track record, the credibility, Do they have complementary skill sets? And I think this is really important because this is a team effort, and you've got to bring in people who have complementary skill sets, whether it's the core biology, whether it's the medicinal chemistry, whether it's the clinicians. You know, it's a team effort to get anything developed. We look at the enthusiasm and commitment of the team. You know, how committed are they? Because it's a long slog and it's a lot of effort. And how reliable. So if we ask you to do something, do you actually deliver? And if you don't deliver, that really starts to send some red flags. As companies mature, we as an investor will start to recruit in a dedicated C-level management that will really take the company and take it forward into its future stages. But at the early stages, it's going to be the academic. And is the execution plan credible? And obviously we've heard a lot about the steps along this earlier today, but we want to make sure that those technical risks that you see in the program, whether they're around medchem, physicochemical properties or potency or PK, you can address those. Can you achieve the target product profile that you aim to get? Are there enabling assays and tools and reagents available in the lab? Are there relevant preclinical models that you can use to build up your data pack and justify and de-risk the program? And do you understand the potential tox issues that you might encounter along the way? We look at things like manufacturing and formulation. Manufacturing, if you're going to do small molecule synthesis, it needs to have a synthesis route that is viable and cost effective. And then lastly, the clinical development path, which is really, really critical. You cannot develop a drug without first of all thinking about how you're going to do the clinical trial. What sort of patients are you going to recruit? How are you going to recruit them? What sort of biomarkers or endpoints are you going to look at? And these are things that we think up straight off before we even initiate. And this is why I'd really recommend trying to bring in clinicians at the earlier stages so you can help think about these sorts of issues because they're really, really critical in terms of how we look at a potential future program. So drug discovery is not research. It's far more difficult, it's extremely expensive, and it's a very high risk. If you embark on these sorts of endeavours, the path will be bumpy. I do think that there are many rewards along the way, whether you achieve success in the end or not, and I think you certainly will, as scientists, grow and improve, and I think the quality of your research will improve a lot as well if you go down this sort of path. But if I have one piece of advice, and just one, it's talk to people, engage with people, talk to your TTO, talk to investors like us. We're covered by confidentiality with UNSW. We can go and talk to you at very early stages and give you guidance about what we think of as a viable development path in the early parts of this. Talk to clinicians, engage clinicians. I really stress that. Talk to KOLs, because if you have a hypothesis around your target, we are going to try and validate that with KOLs in the field. And talk to industry reps, talk to pharma. There's lots of pharma scouts available here in Australia. They're really great and they're open to talking to you. So I think that everyone wants to support the industry here. So go out there and engage. And as I said, we're with IP Group and we're very happy to engage with you. And if you have a project, please come and talk to me.
Our next speaker is Associate Professor Orin Chisholm. And Orin will be speaking about regulatory processes.
Thanks, Mary. Thanks, Jeff, and to the organising committee for giving me this opportunity to talk to you today. Just a bit of background about myself. So I have a long history of working in the pharmaceutical industry, in regulatory affairs, getting products registered and on the market with a number of different companies as I've listed here. And I have a strong background also in education and regulatory affairs. So you've all seen this slide multiple times today, but I just wanted to remind you that the process of taking a product through the development stages and onto the market is quite complex, and that you might start off looking at 5,000 to 10,000 compounds, and there's a high attrition rate, especially in the drug discovery and preclinical stages, but also through the clinical development stages of the process, and that you might eventually end up with one approved product. So FDA is the regulator in the US. and that the estimates are that today it costs $2.6 billion to get a new product onto the market. I just wanted to point out on this slide some of the regulatory standards that you need to comply with at different stages. And so a number of our speakers this morning have also discussed that you need to adhere to good laboratory practice, especially for your toxicology studies in the preclinical studies. But once you move into clinical studies, you need to comply with good clinical practice guidelines. The manufacturing needs to comply with good manufacturing practice. Once the product's on the market, you need to comply with good vigilance guidelines as well. And that here in Australia, the TGA is our regulator for therapeutic products, and they conduct a number of inspections. They conduct good manufacturing practice inspections, good vigilance practice inspections, and they've just recently announced that they intend to start doing good clinical practice inspections as well. The stage between preclinical and clinical is where you if you're thinking of submitting to the US regulator, you can submit your IND, which is your clinical trial application in the US. And the FDA gives a lot of guidance to companies and people that come to them for advice about the clinical and even the preclinical studies for developing products to get them to market. In Australia, you mostly would be submitting a CTN application, which is just a notification to the TGA, unless you're developing a class 4 biological molecule, so your gene therapy products, for instance, where you need to submit a full application similar to the IND application that you submit in the US. And then once you're through the clinical stages, the NDA in the US, the new drug application, or your marketing application here in Australia and other jurisdictions is required. That process can take about a year to get a product approved through the regulator, so they're evaluating all of the data that you've collected, so all of your drug discovery information, your manufacturing information, your preclinical studies and your clinical studies. And they look very thoroughly at all of that information to come to their decision on the risk-benefit equation for that particular molecule. But most of you are not at that stage of the drug development process. Most of you are at that early stage, that preclinical stage of drug development, and so your outcomes for your particular research is often going to be out licensing or coming to some kind of partnership arrangement, as Steven's just said, with large pharmaceutical companies that have the money from their investors to be able to take it through the very expensive clinical trials process. In that early preclinical phase, there are a lot of failures, and you'll be hitting a lot of these failures, and I've just listed some of the reasons for these failures in this particular slide. And I just wanted to point out that more recently the two main reasons for failure in early stage drug development is the non-clinical toxicology studies, but also rationalisation of company portfolios. Companies are beholden to their shareholders, and they need to maintain profitability, and if they think your product is in the end not actually going to make a generous benefit to the company, then they will shelve the product and not take it through development. But over time, that preclinical toxicology studies, and there's been a lot of emphasis on those studies earlier this morning, so you really need to focus on those preclinical studies and ensure that you can get the product through that stage. So how are you going to de-risk your product so that you can get it through that valley of death and into clinical trials? So there are a number of goals in the development program that you can take into account and that you should consider, and planning for achieving these development goals and keeping these in mind front and centre will help you de-risk your product. So you need to really sit down and determine what all the go and no-go decisions are going to be, and at what stages you're going to be making those go and no-go decisions during your development. And you need to actually adhere to them. If you've put them down in black and white in writing, then you actually need to adhere to them, not say, oh, I'll just go a little bit further along the pathway you need to be very firm in your decisions at that point. So you need to avoid spending time on interesting observations, and you need to stay on the critical pathway to getting your product to market. You need to ensure that the preclinical studies support your clinical programs, and that you perform studies that are required to allow the initiation of your safety studies in humans. You need to obtain proof of concept, those phase 1 studies, as early as possible. And you need to establish the therapeutic value in the patient population. So we're increasingly seeing that the major hurdle now is not the actual registration process. The major hurdle globally is now the reimbursement process. So being able to explain not just the risk-benefit equation for your product, but the cost-benefit equation for your product. And so there is a major emphasis now on being able to explain the value of your product in the patients that you're trying to treat. So you need to also deliver a safe and effective product. That's your hurdle with the regulators. And AstraZeneca have developed a great tool for looking at this and taking products through the development process. And they've published it, so that's why I know a little bit more about it. So looking at the right target, having the right tissue, the right safety, the right patients, and the right commercial potential for your product. So you need to put all this documentation together to ensure that you are not going to fail in your drug development pathway. And one of the tools that you can use to do that is to develop your target product profile. So this is a tool that is often used by the regulators, especially the FDA, and they have put out a marvellous guidance document on how to use this tool, and it's freely available on their website, and I've put some links in the slides as well. So this is an excellent strategic planning tool, and I would really urge you all to look at using it, and you start using it right at the beginning of your development process. It's a statement of the overall intent for the drug development program. It specifies the labelling concepts, so what indications are you going to do? What claims are you going to have? What's your target population? What specific studies are intended to support those labelling concepts? It maximises the efficiency of the drug development program because it keeps you on track. And it's a dynamic summary document. It changes, it's a living document, and as you get more information and do more experiments about your product, you can add to this document. And this document is a great document to start with going into your out-licensing or partnership discussions with Big Pharma, because it lays everything out really nicely and clearly, and they can see where you're going with the product and understand that you know the value of your product that you're trying to develop. There are a number of different sections, and they're outlined in the FDA guidance document. So it covers things like the indications, the usage of the product, what's the dosage and administration, so you need to understand your formulation for your product. How are you going to deliver it to patients? One thing that hasn't really been touched on a lot so far is you need to go and actually talk to patients. Go and talk to the patient organisations for the disease that you're looking at. You know, if you're developing a product for lung cancer, go and talk to the lung cancer patients group and find out how they are living with the disease, because patient involvement right from the early stages of drug development can also help keep you on track and make sure that you're developing a product that patients can use. If you're developing a product for arthritis and it's an injectable and they have to mix it all up and try and inject themselves and they've got sore joints, they might not be able to do that. So then you need to think about what other ways of delivery you can use and develop the formulation to deliver that product to the patients. So I'd really urge people to actually have those conversations with patients early on as well. So this is just some of the links to the various regulatory guidance documents. There are a lot of guidance documents available to you, and the regulators want to see that you have conducted your experiments according to these guidance documents. And if you don't conduct them according to these guidance documents, that will lower the value of your product to companies that are looking to license in your product, because they're going to have to go back and repeat them all according to the regulatory requirements. So if you can do that up front, work with organisations that have GLP standards and accreditation in place to do your toxicology studies, your DCP studies, make sure you're adhering to those requirements. That will make it much easier and make your product more valuable when you're trying to undergo those negotiations with industry. And just in my last couple of minutes, I just wanted to point out a couple of tools. So at UNSW, we have a database available through the library. It's called Biomed Tracker. And this is a really useful database, and I'd really urge you all to log in and access this database. You can search the database based on disease areas, products. You can see what products are at what stages of development. It looks at the whole commercial development of products that are on the market and in development, and it's updated routinely. So you can see if you're looking at products for solid tumours, for instance, you can see what other products are out there, what products are in development for solid tumours, see where your competition is and help frame your target product profile based on some of this competitive intelligence that you can obtain from this particular database. And I can't see Jenny here now, but she was here earlier this morning. So Clarivate have a number of tools, and I think they've been in discussion with people, some of the people here in the audience, about trying to have access to some of their tools for early stage drug discovery and development. So there are just a couple of tools that you might be interested in looking at or having discussions with organisations to access those tools. And just a plea that this is a process that's, getting a product to market is not something that is conducted in isolation. You really need to work cooperatively and positively with industry, with government, with academia, so that we're all on the same page. Everyone wants to benefit patients in the long run. So thank you, everyone.
Our last speaker before the panel session is Dr. Charlotte Lemech, and Charlotte will be speaking about early phase clinical trials.
I'm the Medical Director of Scientia Clinical Research, which is a dedicated early phase trials centre located at Prince of Wales Hospital, and I'm also a medical oncologist there. So thank you for the invitation to talk today. So I just wanted to give you an introduction to Scientia, as not all of you may be aware of this Phase 1 centre that's available for collaboration for researchers, pharma, through that early phase trial process. So it's the first purpose-built, publicly funded early phase clinical trials facility in New South Wales, and it's really designed to facilitate phase 1 trials across all disciplines. So I'm a medical oncologist. We do a number of oncology trials there, obviously, and these are both targeted agents, a lot of immunotherapy and different combinations that are coming through. But we also work with university investigators and hospital investigators to facilitate trials across other disciplines. And this has included endocrinology with diabetes studies, working with hepatologists in NASH studies and other hepatic diseases, working with renal physicians, renal transplant studies, nephritis, those areas. And we really aim to be broad in the groups that we work with to try and get these new drugs tested safely in working in both healthy volunteers and also in patient studies, just depending on the drug that's coming through and the best population to test it in. So it's a controlled entity owned by the university and funded initially with university, Commonwealth and state funding. And it's located within the Bright Alliance building, which brings together different cancer services at Prince of Wales Hospital. So the Naloon Comprehensive Cancer Centre is an adult oncology unit, and there's also space for Sydney Children's Hospital based there. As an overview of what we have on site, it's both an inpatient and outpatient facility, and this includes a 30-bed ward. So depending on the type of phase 1 trial that we're conducting, there may be purely outpatient visits, which are done in a long day, for example, with the intensive PKs that are sometimes required, and I'll go into that a bit later. But there's also inpatient aspects that are required, so particularly for higher risk drugs where you might be concerned about infusion reactions or with the immuno immuno-oncology and stimulation of the immune system acutely, you can get something called cytokine release syndrome. Then there may be up to 3 or 4 days of testing in patients, and we keep them on site during this period to monitor and treat these reactions as required. So we have a 30-bed ward for this, which is made up of a 24-bed open area, but also 2 and 4-bed private rooms. So, for example, we were involved with the Parkinson's disease study where it was preferred to keep those patients coming along with carers away from the bulk of the treatment area where we had a higher flux of healthy volunteers and other patients coming through. So we were able to offer more private areas to accommodate different patient groups. We also have an IP suite on site, so for storage and preparation of investigational product, laboratory for specimen processing and storage, so particularly with the PK and PD specimens that come through. And the picture there is just of a small section of our ward area. So none of this is rocket science, but there's a few key components of phase 1 clinical trials that I thought I'd just highlight. So you'd already be aware that this is to assess the safety of a new compound, and I think the majority of today's talks have focused on that drug discovery and toxicology that gets down in the years prior to coming to phase 1 trial. And we're essentially looking to define the maximum tolerated dose and recommended phase 2 dose. So things that have probably already been covered today, assessing the starting dose. So what dose are you going to select based on the toxicology data to bring into the phase 1 setting? And there's a number of different algorithms for working this out. Different algorithms are used depending on whether you're doing healthy volunteer or patient-based studies. So for example, the NOEL or the No Observed Adverse Effect level is based on your essentially your non-rodent and rodent studies. So you want to do your toxicology in 2 different species, looking at what dose levels that you start to see adverse effects, and then calculating your human equivalent dose to take into that first in human, first in patient study. Sometimes you also may use a HNSTD, so the highest non-severely toxic dose, and this is more commonly used in the oncology studies. And importantly, particularly for some of the immune stimulatory drugs, looking at the minimum anticipated biologic efficacious level, so the MABEL. And particularly for some of the T-cell agonist drugs that have been developed, it's important to look at that biologic dose rather than purely at toxicology alone, as it may provide a safer starting dose level in humans when your preclinical and your animal models might not truly reflect what happens in the patient setting. Based on your preclinical studies, there may also be particular toxicity that we need to watch out for when we move into the patient setting. So whether this is cardiac or eye toxicity, for example, then we need to take those factors into account when screening patients for the study, and also considering management algorithms for if we do come up against this toxicity. Depending on your class of drug, there may already be data in this area if you're developing in an area where there already may be similar drugs available, so in the immuno-oncology space, for example, but there may also be different toxicities relevant to your pathway. The historically phase 1 trials have been looking to escalate in small patient groups, so it's the classic 3+3 design where you're putting 3 patients on at a particular dose level before you move up to the next dose level and so on, and you're looking to escalate until you reach the dose-limiting toxicity. And this is something that's important to define in your synopsis and protocol development. And there are standard dose-limiting criteria that apply, but there may also be some that are particular to the class of drug that you're developing. an important part of the phase 1 clinical trial is really gathering that pharmacokinetic and pharmacodynamic data. So looking at the drug levels in the patient setting, and this is where you may have patients undergoing 12 hours of PK testing the first time that they're dosed with the drug in the unit. So it is a big commitment that these patients and volunteers commit to, and it often goes up to the 24, 48 hour and then up to 3 weeks thereafter, just depending on the agent and its half-life. And I think the pharmacodynamic data and how to incorporate that into your phase 1 trial is key, and increasingly we're seeing that play a role in dose selection, so that we're not just increasing the dose on these trials to get to toxicity, but we're incorporating that pharmacodynamic data, so the T cell response, for example, inhibition of certain target pathways, to determine earlier on, is this drug actually hitting the target that you've designed it to hit? Because if we're there may be doses that you reach on a study that you predict preclinically will have shown activity, but if we're not seeing that in the PD data, then it may be an earlier time point that we consider whether this drug should continue to be escalated or not. So the synopsis is really the backbone of a phase 1 study, and there will be a number of factors which have already come out of the preclinical work, which will come into the synopsis. But coming from the research setting, and again, as has been mentioned before, it's important to get the opinion leaders in your area and clinicians involved early to help guide this development and make it relevant for the patient group that you're looking to develop the drug in. And I think talking to patients early, as Orin has mentioned, and talking to patient groups is also key in this area, particularly when you come to the part of the synopsis and protocol called the schedule of events, which is how often you're bringing your patients in to have this testing done. If you have an intravenous agent that needs to be given twice weekly and takes 4 to 6 hours to give, then this is going to be a much harder group to recruit or trial to recruit to, compared to a 3 weekly dosing, for example, or oral agents. And these are all things to take into account when trying to decide on that schedule and formulation. So objectives and endpoints. So essentially for phase 1, you have your primary endpoint looking at the safety and tolerability, determining the maximum tolerated dose or recommended phase 2 dose. Your secondary endpoints looking at efficacy and some of that pharmacokinetic data. And I think the exploratory endpoints are important in looking at that biomarkers and PD data early on in that phase 1 setting. So the example of larotrectinib was given, where you have that biomarker early, which is really guiding your the group of patients that you're recruiting to, and it's for trials where you may not have a clear biomarker to start off with. You want to get tissue, blood samples to try and work out at an early stage what may be relevant biomarkers for your particular agent. So working out the patient number and what type of sites you'll be using. So for the first part of phase 1 studies is really that 3+3 dose escalation, and increasingly we're seeing large expansion cohorts, that the phase 1B, so to speak. So over the last few years, rather than having your classic phase 1, 2 and 3 study design, we're seeing more of that phase 2 trial come into phase 1. And it's these expansion cohorts in phase 1B, where you might have pre-selected tumour types which you're looking to expand in. I think it's important to write the protocol in a flexible way, as there may be certain areas that you have focused on in the preclinical development, but when you get to the clinical setting, it's interesting to see where trials can take a turn, and you often in the solid tumours, you'll enrol all solid tumours to start in your different escalating dose levels. And it's interesting to where relevant, include tumours that might not be targets for the expansion, for example, because you can see signals in tumour types that you may not have been identified preclinically that have then changed the direction that a trial has gone in. Eligibility criteria, making it relevant for your patient population that you're looking in. I mentioned the schedule of events and keeping that accurate, it really helps with budgeting. And again, so talking to your clinicians about what investigations at what time points are required can help you develop an accurate budget when you're looking at funding as a whole for going into phase 1. Just being aware of time. So I've talked a bit about how we've seen more expansion cohorts and greater patient numbers come into that phase 1 setting. So rather than just the historical 20 to 50 patients seen on phase 1, more often see between 100 and 200 patients that are planned to be enrolled based on the success of that initial dose escalation. And for drugs like pembrolizumab, when Merck did their phase 1 study, that phase 1 ended up having 1,000 patients through its different expansion cohorts. In terms of the PD work, again, this is relevant for oncology, but incorporating, where possible, the pre and post-dose tumour biopsies and liquid biopsies where that can be included. And often where possible on progression can be relevant, as this can change the direction that combination therapies might go in thereafter if you've identified a particular mechanism of resistance. An example of that is with the BRAF inhibitors in melanoma. Some of those early studies include those sequential biopsies, and they found other parts of the MAP kinase pathway being relevant with resistance, and so subsequent combinations utilised that scientific rationale. And also importantly, using those PD parameters to help define the biological efficacious dose. And taking into account that if you're not seeing the PD outcomes that you'd expect, then to discuss that with your safety team and researchers about the further continuing the study. But also if you are seeing biological efficacy at lower dose levels than what your escalation might take into account, then to consider how high you escalate, because there obviously is the risk of greater toxicities as you escalate further, and just making it relevant to the efficacy that you're seeing. Thank you.
So if we can please introduce ourselves for those who haven't spoken.
I'm Michael Wallach from UTS. I'm here in my role as director of the SPARK program for Sydney and for Australia, and also globally for translation and medical research.
Phoebe Phillips, University of New South Wales. I'm here representing the cancer theme and CAG, and also recently did an Eisenhower Fellowship travelling across the top entrepreneurial hubs in the US.
My name's Steve Palmer. I'm the Senior Business Development Manager for UNSW in the Life Sciences, so I work for the Tech Transfer Office, and I'm standing in for David Morris, who couldn't be here today.
And I'm Anita Vandermeer from the Office of Health and Medical Research, New South Wales Ministry of Health. I'm here today for Clinical Trials New South Wales, and happy to answer any of your questions after the panel today on clinical trials in general.
Thank you. So I think I'll start with Michael Wallach. In the first session, I think it was Professor Edna Hardman that mentioned Spark Stanford. So Michael, can you speak to us a little bit about the Spark Global program, but also Spark Oceania and what it does to support commercialisation and translational research?
So Spark at Stanford started over a decade ago alongside a program called Biodesign, where Biodesign was focused on medical devices, Spark focused on drug discovery. In those early years at Stanford, people started to take notice of the fact they had an enormous success rate as measured by entry into clinical trials and licensing of their projects, where they're running about a 60% success rate, which is unheard of at any other institution. part of the reason for the success certainly are the people who are involved, and in their case, it's the academics themselves. It's not just waiting for tech transfer offices to come and knock on our door, but it was the academics engaging with the process. And it's a 2-year program where meetings are held weekly over a period of 2 years. Because as you're all hearing in these presentations, translation of drug discovery is extremely complex and requires an enormous amount of mentoring and support. In those early years at Stanford, they had present in the room everyone, like sort of in the session, that is involved with any step along that pathway. And it's that constant support that led to their success. It's not magic, it's hard work. And it's somewhat, for a lot of academics, comes into conflict with the need to publish, write grants, and do other things. People started to value this at Stanford. In fact, the NIH started to prefer funding applications coming from Stanford people who are in Spark as a benefit, and believed that the likelihood of success is much greater. As a result, starting about 7 years ago, we decided here in Sydney to emulate that program, and I was selected and volunteered to be the first director. I was minding my own business at the time. Little did I know what I was getting involved with. And since that time, and to make a long story short, I've been involved now with programs that started in Sydney, at University of Sydney, UTS, Macquarie, Calling Institute, now at Monash in Melbourne, and moving to TRI Brisbane, Adelaide and Perth. I'm involved myself personally with about 30 research groups, and we call them Sparkees, not the greatest term in Australia. So, but around the world it's okay. I think there are two aspects, as I mentioned to Phoebe this morning, to Spark that's really important. One is academics really taking responsibility, so social responsibility, as was being said before, to be concerned with our patients. And we must keep that in mind all the time. The second aspect of it, I think, is the constant support we give to our Sparkees. It's that feeling that you're not alone. As a single institution or a single group in a single institution, it's almost insurmountable, even with the IP and other groups helping. It's an insurmountable challenge that's very hard for any single group or institution to overcome. Even at Stanford, it's extremely challenging. So part of the idea of Spark, which brings us to national and international, was to create Spark Oceania under Accelerating Australia, funded by MTP Connect. And then we recently formed Spark Global. And from the time that I started the program together with Stanford about 7 years ago, we're now up to 23 countries all around the world working together. And through that network, if somebody comes up with a good idea here in Sydney, the idea is to globally work together to help each other to be successful. So that's in a nutshell.
Excellent, thank you. So I'd like to open up for questions, please.
Hi, and this is for anyone on the panel. I'm just wondering about toxicity and what is acceptable. If you're seeing a therapeutic index of 100 or something in vivo in my studies, is that going to be too much of a risk to go to clinical trials, or that put people off? Like, what is acceptable in mouse models? No one?
Perhaps someone in the audience knows. Yeah.
I can make a stab at it, but what you're the clinical trial? No.
Oh no, we have to add our comments together. So, in your preclinical models, I guess we're looking at the toxicology studies where different dose levels are tested, and what adverse effects you're seeing in those animals. What organ system and autopsies affected? Were there animals that died at the certain doses that you were giving to them? And then, so it's not particularly answering your question about the therapeutic index, but it's essentially you're looking at what milligram per kilogram dose you can give to the animals in your tox studies. Again, using those two species, so both rodents and non-rodents need to be looked at. So there's often the monkeys or dogs as your second species that's used, and then looking at what safe dose level can be given without those severely toxic effects being seen in the animals, and then based on those, so it's often 3 different dose levels, or that I've seen most commonly tested in those GLP tox studies, and then based on that dose level, you then do work out your human equivalent dose that can then be taken into human and patient testing.
Can I just add something else? It's going to depend on what the indication is that you're looking at, what patient population you're going to be giving your product to. Is it something that's going to be administered chronically to the patient over a long period of time, or is it just a one-off dose that the patient's going to receive? And also what kind of disease it is, because you're going to have a look at the benefits over the risks, and have higher risks associated with things, diseases such as cancer, where there might not be any other alternatives available to the patient, compared with something else that you're looking at.
Just add something to that, because I think what we've heard a lot today is people thinking about the end first and working back. There's one compound that's a glutaminase inhibitor, CB839, that's 200 mg per kg twice daily oral. As long as the appetite is there for that drug in a patient population, I think the hurdles in an animal model can be overcome. But if you haven't thought about the target market and haven't got the push behind it and the money behind it to push it through, if it's not a very good drug in mice, it's probably never going to make it through. So I think that's a lot of what people have been talking about, is thinking about the end product. If there's an appetite for the end product, then some of these, it's a really awful drug. It comes out a solution that it's not bioavailable, and yet somehow still it's in the clinic because there's an appetite for it as a target. So, I think that's probably important.
My question's to Michael. Thank you very much for that detailed description of Spark, and it sounds very exciting. And please excuse my ignorance. I've probably spent a little bit of time recently just being an academic. But with your presence here today, are you looking at setting up a Spark program at UNSW, or if those of us who might be interested, are we to go to UTS or USyd, or?
So we had a meeting this morning.
Okay, good.
And what I said and what I'll repeat is, in my role as director of Spark in Sydney, I don't distinguish between institutions. We want to see all institutions join the program. We want to see everyone benefit, and we'd like to see the program starting as soon as possible, and I'm happy to facilitate the program as long as you need me. I've been going back and forth to the University of Melbourne for over a year now. I'm happy to go back and forth between UTS and UNSW.
Okay, no, you'd be most welcome, certainly. And just a bit, if I may, a follow-up. I'm assuming that probably at the Stanford website or on the Spark website, there's a list of highly relevant examples of just the success of the program.
There are. We recently had the Spark Global meeting here in Sydney. We have a pamphlet that describes a small portion of our projects. Spark has already led to saving thousands of lives around the world. We're very proud of that through some relatively simple things. Stanford very much focuses on repurposing of existing drugs to have faster impact on patients. Pharmacogenomics is a big area that we're very interested in, and many other things. So I'll do anything I can to facilitate you to be part of the program.
And just on that point, following my discussions, I ranked the Spark program in the top 2 in all my travels around the top entrepreneurial hubs across the US. And certainly I'll be leading those discussions at UNSW. So if you have an interest or want more information, definitely email me.
I have a question, and maybe for Orin, but whoever else on the panel can answer this. So with respect to drug manufacture for phase 1 trials, I understand that this TGA has different levels of acceptability compared to FDA. So FDA, you have to have GMP quality. You have to have GMP manufactured drugs for phase 1. But TGA, no. Is that still the case? Can be GMP-like.
Yes. So if you're looking at a global development program, you need to do your research and your work according to the strictest guidelines that you need. So if you're thinking of putting your drug into patients in the US and the rest of the world, then you probably should be adhering to the GMP requirements.
On the other hand, I've found that when companies approach you and they're interested but they want phase 1 data, and you struggle going, I haven't got the money, I haven't got $1 million to make this drug under GMP, but they want phase 1 data, and so you get phase 1 data and then they're interested. But I guess you may still go backwards because it's not going to be acceptable by FDA. I mean, you're in it, you know what I mean? It's that kind of you're damned if you do, damned if you don't situation to go forward.
If you can negotiate and get the money, I would do it under GMP conditions.
Alternatively, taking the investment dollars into account, Australia ranks within the top 10 in the world for clinical trials, and we do a large proportion of the global early phase clinical trial footprint here in Australia. Having your phase 1 trials here in Australia and in New South Wales will provide you with the data which will be acceptable to the FDA and global multinational companies without needing to have strict GMP adherence to your product.
So that confuses me.
I'm happy to talk about it a little more offline.
Just let me add to that a bit from colleagues at Stanford, which is also important to remember. If you go out with your phase 1 trial with a given type of drug product, that you don't make it too pure, because it will be difficult to manufacture at that level of purity. That's counterintuitive, as I see in your face. And I found that myself working at a pharmaceutical company. You produce something at a very high, extremely high level of purity because of that inclination to want to be perfect. But in fact, if it cannot be produced and manufactured at that level of purity, the clinical trial becomes irrelevant.
Do we have any other questions? Yes. Okay.
Hello, my question is for Steven. So I just want to know where do venture capital stand on the nature of the drug? Because it looks like if it's not a small molecule, like it's not going to get funded, or at the moment it looks like if I cannot produce a small molecule, or like if I not transform my drug into a compatible small molecule, it doesn't get much of a chance of getting funded. Is that where the venture capitals are sort of standing, or are they kind of taking the risk of looking at other kinds of drugs, not just the small molecules?
Do you mean other kinds of drugs like biologics, cell therapy, gene therapy?
Yeah, because it looks like at the moment it's just small molecules and maybe a few antibodies, but like, for example, peptides or sRNA or like other kind of type of...
I would say the opposite. I'd say small molecules are probably the most difficult drug discovery type projects to come out of academic labs. If I had, this is probably not the right forum, but if I had advice, I'd say avoid small molecules as much as you can. I'd go biologics any day of the week. From the simple perspective that small molecule drug discovery is extremely difficult. It's very challenging to do a high throughput screen, to get good hits, to then take those hits and do a lead op program and get to a viable lead candidate. It takes a huge amount of money, it takes a lot of time, and the IP that you generate then tends not to be something that really universities are very good at. Universities, in my view, are very poor at doing small molecule drug discovery, whereas on the other hand, biologics is something where the IP is really owned by the academic, and you can do a lot of the preclinical efficacy work, and the same, more or less the same molecule actually goes into the patient. So I think it's a much stronger rationale from an academic point of view to go down a biologic route rather than a small molecule route.
add some numbers. 20% of the drugs approved last year by the FDA were biologicals. 70% of the top-selling drugs last year in Australia were biologicals, and the FDA stated that they're currently receiving around 200 IND applications for cell and gene therapy type products, and they're expecting by 2025 they'll be approving about 10 to 20 products in that category per year. So biologicals and the gene and cell therapy products are growing and accelerating at a very fast rate.
But is that a good thing? So, I mean, we're talking about patients and this being patient-focused. All the biologicals and cell therapies are very expensive. You know, how does this fit, you know, thinking about reimbursements and how are we going to be able to better suit patients, having to have an intravenous injection, having to have cells taken out? You know, it's all well and good to say, well, let's just stick with antibodies. They're much easier to get into the clinic. But, you know, what's the responsibility we have in terms of thinking about how we generate? Do we just go down the easiest path if that's going to work? Or, you know, should we be thinking about this? Is there, if you've got the opportunity for an antibody or a drug, is it better to just go the antibody path?
In short, yes. I see no real... I mean, in terms of target engagement and specificity, you can't beat a biologic. And with small molecules, you always run the high risk of both on-target and off-target toxicities. And that's always going to limit your dose. I think a once-a-day oral is the ideal sort of target product profile, but then you're competing against a once-a-month injection or a once-3-month injection, or as you extend half-lives, you can have, you know, I think from a patient benefit, which would you prefer? To have to pop a pill every morning or have to go into a clinic and get an injection once or an infusion once every 3 months or 6 months? So I think, you know, you really have to look at the particular indication, look at the particular case. There's no one size. Your biologics are going to be the answer to everything. On the cost, again, the cost of, you know, antibody therapies, you know, in terms of the cost of production and cost of goods sold, is coming down and down and down. So the value and the cost of drug pricing doesn't really bear a strong relationship to the actual cost of producing it, with the exception, I'd say, of probably cell therapies at the moment. What you're paying for is the value. And that's true of a small molecule, which is going to be priced just as high as a biologic if it provides the same level of value.
I'll just add to that from a university non-profit point of view. If you can identify a target and find an existing molecule that works, even though there's no money to be made from it, and that's a good approach. You already know the safety profile, and you start from a good position. So whereas venture capital is looking for potential blockbusters, we're looking for things that work and get to the patient quickly.
Do we have time? We have time for one more question. Yep.
So my question is actually to Michael regarding the repurposing, you know, you have done at Stanford. I guess it's like, you know, because it's repurposing, so the farmer, you mentioned like investor, probably don't want to really put in money into it. So who's going to fund the clinical trials, right? So you can do an investigator-initiated trial, which is fun, but that doesn't count anything, right? So 10 patients, 20 patients, that's pretty much it.
From what I understand from speaking to the Stanford people, funding of these trials hasn't been a huge challenge. They've also do these trials in-house in a way that's very cost-effective. So clinical trials there are not costing $20 million, they're costing $100,000. And so it's affordable, becomes an affordable thing to do. Their ability to get the clinicians to take up these trials, I think, is part of the success of the program. It's like, as was said earlier, keeping close contact with clinicians so that they'll want to do these trials. They've done trials, and we've seen in the media, on 3 patients, and have already had success. So I think, again, there's the VC and investor way of looking at things, but from a Spark way of looking at things. We just want to get to a patient. We don't care if n equals 1. In that case, people bend over backwards to find a way to make it work so that the cost, as I say, is very low. And the effects have been absolutely stupendous.
Okay, so thank you. That's the end of the panel session. I'd like to thank all the speakers and the panel members for their contributions today. So please join me in thanking all of our speakers and panellists.